Oh, it's 10 o'clock. The Garcia program always meets at exactly 10 o'clock in the morning. That's what we've been trying to do now for all the entire summer. Because one of the things that we learned is that we all work together and coordination between what we all do is of primary importance. So welcome to the 2022 End of Summer Research Scholar Symposium. And we had a preliminary symposium only 2 weeks ago. But what happened in the meantime was that what our students learned is that your best data always comes in at the very last minute. You prepare, you learn the field, you learn to work with your coworkers, you hit a resonance, and then the data starts to fly in. So there's a big difference even between the presentations that we had by our students just 2 weeks ago and the quality of the work that they've presented here. But before I begin, let me share my screen because there is one thing that's really important and that you can only see by this screenshot. And again, as I'm saying, it's only the latest data that counts. And you can look at the times over here. And it's not just Romy, it's a lot of people. I have a whole set of these on my phone. I can just grab this in a minute. And guess who all these messages are addressed to? You can see here, the one and only. And this is a typical pose for Jessica, solving somebody else's problem. So before we begin, just a huge shout out to Jessica Hoffler, who put it all together at the last minute. So what you see today is really a masterpiece of work by Jessica. Thank you. Okay, so now we're controlling the screen, so you can take it back again. Okay, we'll start our symposium, the Garcia Symposium today. First of all, as you'll see in the program, the view graph that you just saw before is by Lian Tai, who has made our symposia logo, has won the contest from the other students for now 2 years, 2 consecutive summers. Again, thank you very much, Lian. Okay, so we will begin the symposium today with Mr. Steve Vaccarelli from Brown Brothers Harriman, and also the trustee of the Maureen Charitable Trust that has been supporting the quality of life as well as the research of our program now for the last almost 10 years. Steve, yours.
Thank you, Miriam. As Miriam just said, I'm Steve Vaccarelli. I work at Brown Brothers Harriman Trust Company, and we are a trustee of the Maureen Charitable Trust, a trust that was established about 30 years ago. It was designed to support research into engineering and related physical sciences, and we've been very happy with our support of the Garcia Summer Program since 2005. So we could not be more happy with that. And I just wanted to make a quick comment that to the students, congratulations on a very productive summer. I understand besides all the hard work, you had some fun as well. I'm glad to hear that. And I'm also very glad to hear that you're able to conduct your summer research at Stony Brook in person, unlike last summer's class. I think everyone would agree that there's no good substitute for collaborating in person. So, so I really look forward to your presentations this morning, and look forward to hearing the results of all your hard work. Thank you very much.
Thank you very much, Steve. And our keynote speaker, we usually invite someone from the outside, just like we've been doing almost every day this summer, to get a point of view of research, exciting research going on. And in particular, areas where research directly impacts our quality of life. And one of the best representatives in this area, who has been speaking to our students now, also, like Steve has said, for more than 10 years, since '05, has been Isaac Cohen, Dr. Isaac Cohen from Estee Lauder Companies. Isaac, share.
Thank you, Mary. Good morning, everyone. Can you hear me okay?
Yes, you are good.
Can I share my screen?
Yes.
Let me give that a shot. I'm sometimes technically challenged with this, but let's see if I can do it. Bear with me then, please. No. How do... I'm sorry. I'm just trying to get to my PowerPoint. Let's see. Screen sharing. Okay. I apologize for the delay here. Okay.
I should say, while Isaac is preparing, the Estee Lauder companies have been supporting research with our students and have really had some very, very important breakthroughs, especially in skin technologies. And a number of their people have obtained PhDs or postdoctoral degrees at Stony Brook.
Can you see my screen?
Yes, we can.
Perfect. Okay, thank you. So, Professor Verfelovich, thank you again. Yeah, it's been quite a few years. It's always a pleasure to address your summer scholars, and I enjoy it immensely. I want to wish all of you success as you continue your academic endeavors, as well as when you go on to your professional careers. And speaking of careers, I wanted to say a few words about my own career, how I ended up at a place like Estee Lauder. And in doing so, hopefully we'll answer a question that I know is burning in everybody's mind, and that is, where in the world is the science in cosmetic science? Most people don't think of lipsticks and mascaras and organic chemistry in the same breath, but there is a lot. There's a lot of overlap in a wide spectrum of sciences. And I hope to give you just a little flavor of that today. So first of all, a little bit about me. I was born and bred in Brooklyn, New York. I attended Brooklyn College. I got my bachelor's there. And I had the honor and privilege then of working as an undergraduate with Professor Takanobu Ishida, who has since passed on, but interestingly enough, moved at one point from Brooklyn College to Stony Brook. So there's a Stony Brook connection there. And he was very interested in stable isotopes like carbon-13 and ways of enriching those. I had a small project there on the synthesis of carbon-13 methyl fluoride. It got us a publication in the Journal of Radio-Labeled Compounds and Pharmaceuticals. From Brooklyn College, I moved on to MIT and worked for a professor, William Rastetter, who then moved on from MIT to the biotech industry and got his fame and fortune in California in companies like Genentech and others. Professor Rastetter's interests were in natural iron chelating molecules called siderophores. And there are two examples shown on this slide. Enterobactin is a bacterial siderophore that chelates iron through these tricatechol moieties, and mycelium is a fungal metabolite that uses hydroxamic acid residues to chelate iron. Why do we care about chelating iron? Well, there are a number of congenital diseases, and again, I'm speaking probably about 40 years ago. I haven't been up on, the current medical status, but at least at the time, there were issues with children who had basically blood that didn't work. And the treatment for them was constant blood transfusions. Now, the blood transfusions took care of the blood issue, but it introduced a new problem. And that is, with each blood transfusion, the old blood would plate out, and left untreated, these children would quickly die of iron poisoning. So there was a concurrent therapy giving them iron chelation to get rid of the iron. But now you introduced a third problem, and that is, now you made this iron available to all the intrinsic microorganisms in the body, and you had to give massive amounts of antibiotics to worry about infection. So Professor Rastadter's interest was, could we look at these natural siderophores, which are incredibly powerful iron chelators, and modify them, let's say sterically, so that they wouldn't be taken up by these bacterial materials, by the bacterial organisms. And so we did a lot of work, both on synthesis, synthetic methods, total synthesis. It overlapped with, for example, microbiology. I had to grow my own mold to get my cellulonamide. And we also had a nice program with a clinical team at Rockefeller University who was doing animal testing to see if we were really able to modify uptake of these materials and control infection. From there, I spent about a year postdocing at Brown University, and I worked for Professor Kathleen Parker. Another interesting note, she also at one point moved on from Brown University to Stony Brook. So I guess the lesson there is all roads lead to Stony Brook at some point. Again, my work there continued on synthetic methods. We were looking at a class of antibiotics called isoquinoline antibiotics, got some publications out of that. And while at Brown, Professor Parker had a good friend who worked at a company called Givaudan Corporation. Givaudan's a flavor and fragrance house owned by Hoffman LaRoche. And I was able to get an interview there and get a job there, and was fascinated by the sensorial aspect of the chemistry they did, flavors, fragrances, and related materials. And I actually ended up doing a lot of work, not on flavors and fragrances, but on sunscreens, which was another core one of their interests. And because Estee Lauder uses sunscreens in many of their products, I got to learn about Estee Lauder. I actually was involved in some technical consulting for them on some of our sunscreens, and eventually applied there for a job and moved over to their innovation, corporate innovation group at Estee Lauder. So that, in a nutshell, is how someone who was pretty classically trained in synthesis and synthetic methods ended up moving to a company whose major business is lipstick and mascara and eyeshadow. And I will tell you, when I first started at Estee Lauder, I didn't know the difference between a mascara and an eyeshadow. I do today. I did have to learn along the way. But what fascinated me was the amount of not just science, but scientists involved in Estee Lauder. And Professor Rafelovich mentioned Estee Lauder and Stony Brook, and particularly your program, have a nice long history. There are students that have gone back and forth. We still have a very vibrant collaboration, I know, that goes on. And a lot of, there's a huge amount of not just science, but scientists at Estee Lauder. In our Melville facility alone, there are, I believe today, probably close to 400 scientists. Many of them are PhD level. And that's one of 5 or 6 scientific centers at Estee Lauder. We have centers up in Canada, in the Midwest, in England, in Asia, in France. And I was fascinated to learn how much science really goes on in these products. So just to dig a little deeper in that, and again, hopefully to get back to the question of where is the science in cosmetic science. And let's take a look first from the more hard science side, okay? So obviously, all kinds of chemistry are involved in the development of our products. Organic chemistry, and when I say organic, I mean organic in the way we normally use it as carbon-based chemistry. Many of the materials that go into our products are hydrocarbons, carbohydrates, and so on, but also organic from the sense of life-based materials. We do a lot with plant extracts and so on that have some unique and exciting skin benefits. There's a lot that goes on in the world of inorganic chemistry, especially in makeup products, and I'll say more about that in a second. Biochemistry, obviously, there's a lot of our work at its simplest level involves the interaction of products with skin. There's biochemistry there. We try to deliver various skin benefits like moisturization, like reduction of pigmentation and so on. And we need a good understanding of general biochemistry and skin biology, physical chemistry, because our products span a number of different products forms, anhydrous to emulsions, everything in between, biology, physics, and I'll give an example really quickly of optics and how we use optics in our world. And of course, engineering and medicine, because we live in a really interesting world in cosmetics where our products very often do very good things for skin, but because we're not drugs, we have to be careful how we speak about them. And so we're always interested in knowing where our products in terms of their efficacy end and where the medical world starts. But what's really exciting and what I'm thrilled about and how I spent more than 30 years of my career at Estee Lauder is the interaction between what you might call the hard sciences and the softer sciences, psychology, sociology, anthropology. At a very basic level, skincare and makeup products are very much about how you look and how you feel. Books have been written about the psychology of beauty and what we consider perfect skin, what we consider beautiful appearance. Sociology from the perspective that it's not just about how you look and feel, but how you look and feel to other people and the cultural aspects of that. And anthropology, because there is much that's been done about the cultural aspects, the demographic aspects. Very often we will look at older societies, tribal societies and so on, and how they use plants, how they use colors in their rituals. And that interaction between the psychological aspect and the hard science aspect comes up every day. And it really makes working for a company like Estee Lauder incredibly unique and exciting. Digging a little bit deeper, again, as I mentioned, organic chemistry from both a carbon and life-based perspective. In organic chemistry, iron comes up in all our makeup products. Iron oxides are the workhorses of the pigments in our makeups. Titanium as well. Titanium dioxide is important for makeup products and for sunscreen products, which we work in as well. There are organometallic materials based on manganese, chromium, and others to give us beautiful shades of blues and greens. And for the past 30 years, silicone has been a very important part of both skincare and makeup products. Silicone polymers, silicone spherical materials, silicone volatiles have all really transformed the cosmetic industry. There's another transition going on now. People are moving away from silicone, some of them because of green concerns and so on, environmental concerns. And similarly, we've done work with fluoropolymers over the years as well. And as I mentioned, physical chemistry, biochemistry, the list can really go on and on. Engineering, of course, we don't just make batches, a few hundred grams in a lab. We make multi-tons of materials. We're a $16 billion company, and all aspects of engineering come in, both from a perspective of process development, scale up, product development. We need engineers to help us understand the mechanics of formulation. And importantly, analytical chemistry is a big part of what we do, because a number of our products are OTC. These are, for example, sunscreens, acne products, and so on. And so we need to have very rigorous analytical protocols to support what we're doing in that area. At the end of the day, consumers love our products, and they buy them for pretty much three attributes. One, they like how they feel. And what drives how they feel from a chemical perspective are various oils. Again, those can be hydrocarbons, they can be silicone-based oils. Waxes become important, certainly in our solid products, lipsticks, for example. It may surprise you to know that lipstick chemistry, chocolate chemistry, and crayon chemistry really have a lot in common. And over the years, we've done collaborations with companies like Nestle and Crayola for that reason. Emulsifiers, many of our products are water and oil, oil and water emulsions, multiple emulsions. And the way those emulsions hold and the way they break on the skin affects how they feel. And fillers are often used to modify viscosity and the way a product interacts with the skin. How a product looks obviously is important, not just for makeup, but for skincare products. Pigments drive much of that. Pearls, and the general difference between pigments and pearls is pigments create color through absorption, pearls more through interference. We get some beautiful interference effects using pearls. Sphericals are involved in things like soft focus and light scattering. And then finally, a consumer is concerned with how the product wears. And this is the purview of polymers, hydrocarbon polymers, silicone polymers. And just as importantly, when we design these materials, is not how long they wear and how they stay on the skin and don't transfer, but how easy they are to take off. And there's a whole science based on how can we modify these polymers so they stay on when we want them and they come off when we want them. Plasticizers, because very often these polymers are brittle and need to be modified so they're comfortable on the skin, and volatiles. And again, a lot of work has been done over the years with silicone-based volatiles. We're moving slowly into more hydrocarbon-based volatiles that are carriers for these polymers. And this interaction between how the products feel, look, and wear is what really altogether drives how the products perform on the skin. And that, at the end of the day, is what we spend our time doing, making high performance makeup, skincare, sun care, fragrance, hair care products. How am I doing on time, Miriam?
1 minute.
1 minute. All right. I'll go through this quickly. Just a real quick sampling of how optics impacts our work. Those of you may be familiar, Professor Adelson's the professor of vision science at MIT. And if you Google him, you'll see some really neat optical illusions. What's exciting about this one is if you ask the question, what's darker, A or B? And the answer really is it depends, because from a perception, A looks darker. We all look at this. A, of course, is surrounded by dark, surrounded by light. B is light, surrounded by dark. But in fact, from a scientific perspective, if you isolate these, they're exactly the same color. And you can do the same, and I don't know who the author for this one is, but if you look, for example, at this Rubik's Cube, the dark brown square on top, the bright orange one on the side, again, if you isolate them, are the same color. And the reason this comes to bear, for example, in our world, is we do a lot of work on reduction of skin color imperfections. And one of those areas is under eye circles. People of all ages and all cultures suffer from dark under eye circles. And one of the things we've learned is it's not just about correcting those circles, but modifying the perception of those circles. And we can do that by playing with light and optics. And very often what we do is just blur that interface between the dark under eye circle and the surrounding area. And that will lead to an immediate perception of reduction of those under eye circles, much like changing the surrounding area around those squares changes the perception of whether they're darker or lighter. I'm going to pass through this. I was going to give an example from an optical perspective how we do this, but suffice it to say, we do employ interesting optical materials. We play games with refractive index to blur and create multiple images of, for example, dark spots under the skin, and in that way reduce the perception of skin imperfections. We do that with sunscreens. We play the game sort of in reverse, and we can make more transparent sunscreens that will look more transparent and more aesthetic on the skin. And so at the end of the day, it's an exciting field. It's a nice interface between hard science and perception. And the thought I want to leave you with is, if this world was really designed around black and white, it would be a kind of pretty drab place. And Estee Lauder does, I think, a wonderful job of bringing some color into our life and making our life much more better and much more enjoyable. So I want to thank everybody for their attention. And Miriam, thank you again for inviting me. It's been an absolute pleasure. And I wish everybody good luck as they move forward.
Thank you very much, Isaac. And we have like half a minute if there are any questions for Dr. Cohn from the audience. Just speak up if you do. Okay. And if there aren't,
One quick question, Dr. Miriam.
yes,
Very nice talk, Dr. Cohen. Very nice talk. I just have a quick question. So in your company, like what I'm asking from a biologist point of view, what models you guys use? So skin models I can think of, or any other models you use to like test your products on?
Do you have a few hours?
No, just quickly,
Absolutely. There's a lot of work. Obviously, our company and many of the other cosmetic companies have moved away from animal models.
Right.
We do a lot of work with in vitro models. Some cases, those are purely synthetic models. We have plastic skin models that we use, for example, for France cells and look at delivery studies. Some of those are living skin equivalents. We do a lot of work with ex vivo skin, cadaver skin very often, to look at penetration, to look at delivery of molecules through the skin. So yes, we have, and in my own group, there are 5 scientists strictly focused on delivery science around skin models. And we're always looking for not just biological skin models, but textural skin models. We do a lot of work, for example, with silicone casts that duplicate lines and wrinkles and imperfections in the skin.
Okay. Nice. Thank you.
You're welcome.
thank you. And I should say, Isaac, I still remember the first time you spoke to our kids and showed them how dimensionality, like nano, can change the color of light. And I still pull out a $20 bill. Well, I show them one of the 20s was Estee Lauder nail polish.
That's right. That was, yeah, you know, it's funny you say that because yesterday we were driving down the block and we passed this car that turned from green to blue. And I was telling my son who was in the car with me, I was at that company and we worked on those pigments. So we do a lot of fun stuff.
That's right. And it was inspired by the back of a fly, if I remember correctly.
Yeah, all the interference colors. That's right.
Yes. Again, thank you very much. And
Pleasure.
Our next group would like to, the way that Garcia Symposium is structured, we have students that have submitted abstracts. Now, our next symposium is something very, very special. Not only is Garcia concerned with science, but they are one of the tenets of the program is concern of the ethical implications of the science that we do. And in that respect, we are inspired and guided by Professor Brooke Ellison. And Brooke, are you on? We'll have her, she's around and she may be on shortly. Professor Ellison has been an inspiration to us for many years. And in the last 2 years, she has actually started a journal that deals with different ethical topics run by the students. And I will invite today Maytal Kels, who is the editor-in-chief, and these are the 4 editors of this journal, to explain to you what this is, how it's done, and introduce this session. Maytal?
Thank you. Hi, everyone. I'm really excited to share a project that Garcia students this year have been working on, and that's GESTI, the Journal of Ethics in Scientific and Technological Innovations. I, along with Connie, Navia, and Anya, are the editors-in-chief, and we're so excited to collaborate with Garcia students on 17 essays regarding various scientific ethical dilemmas. And these topics include things from privacy, genetics, aging, sustainability, government, geoengineering, vaccines, stem cells, drugs, and more. And today you're going to hear from the 4 highlighted groups, which you can see on the slide. I would like to thank our mentors, Dr. Brooke Ellison, Dr. Miriam Rafailovich, and Jessica Hofflich for their constant leadership and guidance they continue to give us on a daily basis.
Hi everyone, this is our project, A Salty Situation, is desalination an ethical solution to water scarcity? As global warming causes a lot of droughts around the world, many have turned to desalination. Desalination is a feasible solution, but it leads to a lot more environmental problems. Some of these unintended consequences include dumping the byproduct brine from reverse osmosis plants back into the ocean and increasing fossil fuel usage. So, the increasing popularity and controversy regarding desalination begs the following question. When is it ethical to adopt desalination programs despite potential long-term damage to the environment?
So we consider the ethics of desalination from two perspectives. First, we consider from utilitarianism, which is the principle that says we should promote the maximum happiness for the most people. And that suggests that we should expand desalination to the most vulnerable populations in the world. But on the other hand, we also consider environmental justice, which calls us to provide equal treatment of people with regards to environmental laws. And from that perspective, government decisions need to take into account the harmful repercussions of desalination on the environment. And we also considered solidarity and keep in mind that not everyone has equal access to desalination.
And so after weighing opposing arguments from these sides, our group had decided that desalination should only be used if absolutely necessary. But how do we know what is absolutely necessary? Well, Bentham's utility calculus can be used to determine if, where, and when desalination projects are justified. For example, we believe that if desalination is not the only viable option for making clean water for a given region, alternatives should be chosen. And with that, we encourage you to further explore this issue when our article comes out. So thank you and enjoy the rest of the symposium. Good luck.
Hi, our project is on gene therapy and making a person better or making a better person. So as seen in the figure, gene therapy has grown in popularity over the past few years, and the projected popularity of gene therapy treatment is increasing exponentially. Before we discuss the ethical considerations of gene therapy, let's begin with a little bit of background information. Gene therapy began in 2015 when a human embryo was edited with CRISPR for the first time in an attempt to fix a gene error that caused a blood disease. They were abnormal IVF embryos and they were soon destroyed with no attempt to create a child. The resulting paper was rejected by top Western journals on the grounds that didn't follow ethics rules. In 2018, a human embryo was genetically edited for the first time, and the world's first gene-edited babies were created, which sparked a debate over germline gene editing, as gene therapy and genetic editing raises a host of ethical issues.
So ideally, gene therapy is projected to solve multiple issues associated with harmful genetic mutations, but its scope is limited by the numerous ethical questions that we need to answer before continuing to pursue research in this field. Firstly, it's important to come up with a bright line or a set of parameters for gene editing, i.e., where do we draw the line between creating healthier human beings and improving or enhancing subjective characteristics of humans. Without limitations, it's possible that gene therapies can reinforce existing beauty standards and foster renewed belief in racist and eugenicist perspectives under the guise of creating the so-called perfect human. Because gene therapies are also wildly expensive right now, their use fosters questions about accessibility. If it's only available to members of higher socioeconomic status, the gaps between different socioeconomic classes could also be widened.
So with ethical considerations in mind, gene therapy requires strict restriction. It must be considered that with this expensive treatment, there's an inevitable outcome of widening the currently present socioeconomic gap. In addition, allowing for people to decide while editing genes which traits are to be seen as more desirable will lead to increased societal division. In reality, gene therapy is only ethical in life-threatening scenarios and should only be implemented when no other practical treatment options are available. In these situations, gene therapy should be made widely available through insurance programs and subsidization to ensure that those who need it are able to receive the most effective treatment possible. Thank you for listening. Enjoy the symposium.
Next page, please. Sure, thank you. So I will be presenting our group's ethics research topics. So strategy.
We can't hear you.
Oh, right, I have an audio problem. Can you hear me now?
You're coming in very low, Ivan.
Let me try to
Better.
check my audio right now. I'm basically shouting at my computer right now.
Okay, keep shouting. You're good.
Is this better? No.
Yeah, it's a little better.
Sure, I'll probably have to. I'll just shout at my computer.
Okay, Ivan is
So-
shouting to us from Shanghai, so it's pretty good.
Yeah. Stratospheric aerosol injection, or SAI, is a developing geoengineering technique to mitigate climate change. Research of SAI is carried out with indoors methods including computer simulation and investigation of analogs, such as volcanoes, as well as field methods ranging from small-scale proof-of-concept tests to large-scale climate response tests. During the design of field experiments, a range of ethical issues emerge. As computer models are often limited, the impacts of field research can vary greatly with geographic and time scales, and assessment of risks and benefits are often difficult. We decided to approach these issues from the perspectives of the commonly used utilitarian approach, principles from the Belmont Report, and the concept of risk knowledge calculus. With these, we propose 3 solutions to improve SAI field research design: increasing the types of indoors research methods used, using indoors research to evaluate the necessity and risks of fieldwork, and to implement inductive risk analysis. Thank you.
Yes.
Hi everyone, for our group's ethics project, we examined the influence of federal funding on scientific research. In 2019 alone, the United States government spent $138.9 billion on research and development, which you can see on the graph on the slide. Federal government is the one represented by the green line. We chose to focus specifically on the federal government because of the unique pressure that the federal government can exert with its funds, the ability of the federal government to suppress information possibly in the name of national security, and the higher standard that most people generally hold the federal government to. Therefore, we investigated what the ramifications are once the government gets involved in research.
Specifically, we looked at 3 different case studies, first being facial recognition, a common government tool that's associated with fears of invasion of privacy, governmental overreach, and biased data interpretation. Then we looked into bioweapons. The US government often contracts private companies globally to engineer weapons, and a significant issue is the lack of government transparency. Should the government tell the people what they're working on to ensure that they can keep themselves safe, or keep it a secret in the name of national security? And lastly, biased university reporting. Governments tend to already expect insightful data during the grant applications, meaning that researchers may only gain funds if their project looks promising. And in order to avoid these dilemmas, researchers have previously faked data and results to gain funding for their projects. We looked at each of these issues through various ethical lenses and formulated an argument of how we believe they should all be approached. Thank you.
Okay. Thank you very much.
So I'd just like to jump in and offer my complete commendation for all the work that you guys have all invested in this. I was so thrilled when I saw the number of students who are interested in these kind of really complex questions, right? Scientific exploration in and of itself is a difficult and taxing endeavor. But then when you look at the ethical questions and the socio-political implications of the work that you do, it becomes really a minefield. And as you have already addressed in some of the presentations that you've given so far, these are really complex, tough questions and involve a lot of thoughtfulness. And the way you have embraced these questions, I think, is just awe-inspiring. So I'm so proud of all the work that you have done so far. I'm so excited to read your papers as you complete them. And you have my just tremendous applause for all that you're doing. Thank you, guys. And I look forward to seeing what you come up with. Enjoy the symposium.
Thank you very much, Brooke. And I really should have a shout out to everyone because I could have just as easily put up a screenshot of Maytal's emails to me at 3:04 a.m. last night. And it's amazing that you all pulled this together. Again, thank you so much, Brooke. It's
No, thank you. All the credit goes to the students, and it's absolutely incredible. And I feel very privileged to be working with all of you.
Any questions from the audience? If not, the way that the session is structured, we have all the speakers in one session, and at the end, if you have any questions for that session, please just speak up. Okay, our next symposium is biopolymers, and this will be introduced by Hee-Ting Lo, a new graduate student in our chemical engineering program. And my comment is that the biopolymers that she will be speaking to have exactly the same issues that were raised by Isaac Cohen before for cosmetics. So physics cuts across all disciplines, from soil to lipstick. Hee-Ting.
Hi everyone. So I'm Hui-Ting, and I will be introducing section 1, the biopolymers. So the biopolymers are produced from the rhizobium species strain, which is a bacteria, and the biopolymers is a biodegradable and environmentally friendly material. And this project is a huge collaboration with the U.S. Army of Engineering. So there are several sub-projects based on the biopolymers, and the first one is on the influence of rhizobium species or produce EPM biopolymers on green bush beans, roots, and plant growth. And it will presented by Yo-Yo Zhang, Laura Wang, Sherry Yu. And the second project is on the antimicrobial, antivirus, and toxic properties of rhizobium species, EPS, and it will be presented by Maytal Charles, Alina Samuel, Gregory Rothman. The third project is on the properties and applications of biopolymers, EPS, and nanocellulose fibers with clay in solutions. And the work was done by Rick Chang, Stephen Young, and the last project is on molecular dynamics simulations of soil strengthening nanocomposite hydrogels, and the work done by Jerry Zhang, Vincent Wang, Anthony Jude, and Yoti Shehan. Yep. Thank you.
Hi, today we're presenting our research on the influence of Rhizobium trapezi produced EPM biopolymer on green bush bean root and plant growth. So unlike traditional chemical fertilizers, ethanol precipitable materials or biopolymers are organic biodegradable compounds with an environmentally friendly synthesis process produced by bacteria. The primary goal of our research is to test the effect of these biopolymers on plant root and true growth, which could combat soil erosion.
From bacteria Rhizobium trapezi, two different biopolymers named EPM1 and EPM2 are produced. We treated bush beans with different biopolymers in various concentrations. After germination, we measured the roots and leaves mass of the plant separately and analyzed the stem lengths using ImageJ. The chemical composition in roots and leaves were investigated using XRF. From the data, we find out that all the biopolymer treated groups have a higher mean stem heights than the tap water control group. There is a significant difference between the control and the 50 milligram per liter EPM2 treatment group. The 95% confidence interval indicates that the difference of the mean stem heights of the two groups lies between 7 centimeters to 18 centimeters.
The XRF data also reflects this difference, as the 50 milligrams per liter EPM2 treated group has the highest concentration of potassium and phosphorus in both the roots and leaves, as well as exhibiting the best absorption in the roots for all analyzed elements. However, the higher absorption did not help with root development as indicated by its mass. Both EPM1 treatments groups has the larger average weight of the root system than the EPM2 treatments, with 100 milligrams per liter EPM1 treatment showing a larger positive influence on root development than the EPM2 treatment of the same concentration with a p-value of 0.028. Moving forward, we're currently aiming to investigate reasons why certain treatments are better for roots and other better for leaves. And finally, we want to express our sincerest gratitude to Huiting Liu and Dr. Rafilovich, as well as everyone who made our project possible. Thank you.
Thank you.
Hi. Our group has been working to characterize the EPM polysaccharide biopolymer with regard to its antibacterial, antiviral, toxic, and physical properties. There are many potential applications of our research, including the EPM's ability to strengthen soil and roots and enhance plant growth. With regard to its potential toxic properties, determining whether the polymer exhibits toxic properties or not is crucial to its future use, as EPS-treated plants may eventually be utilized for humans and animals. To test the polymer for potential antimicrobial properties, we performed an agar diffusion assay with E. coli and S. aureus to test on both gram-positive and gram-negative bacteria. For our first test, we used Petri dish testing with tap water and incubated overnight. We didn't observe a zone of inhibition, indicating bacterial growth and a lack of antimicrobial properties. For our repeated test, we utilized a liquid culture containing E. coli, S. aureus, and E. faecalis, incubated overnight. And we observed our results through calculating the optical densities of the various liquid cultures with and without EPM. And we found that the difference between the presence and absence of EPM was not statistically significant. For our antiviral testing, we conducted 2 trials. For our first attempt, all of the cells in both the control and the experiment were killed. And in our second attempt, our control remained alive, indicating that the H1N1 influenza virus failed to infect the cells. We're currently in the process of doing the antiviral testing a 3rd time, and the cells are currently growing.
Our project is heavily focused on characterizing the EPM and how and why it affects the soil in the overwhelmingly positive way that it does. In order to determine this, it is essential to learn the rheological properties of the EPM. A number of tests were conducted using a rheometer, and we concluded that the strength of the EPM increases exponentially with concentration levels, as can be seen by the amplitude sweep graphs and the bar graph showing the highest point of modulus for each concentration. In addition, the yield point where the EPM begins to break and the loss modulus is at its highest point, as well as the delta values, which is the amount of stress the EPM can withstand between its yield point and breaking point, were observed to increase tremendously with higher concentration samples. Finally, we wanted to see if the EPM could recover from being pushed past its yield point. Therefore, we broke the EPM down in the rheometer at its highest concentration and retested 30 minutes later. The results showed that the EPM had values for elastic modulus, which indicated that it was able to make a recovery.
Maytal, we can't hear you.
Maytal, you're muted.
Maytal, we cannot hear you.
Can someone, Eliana, can you take over? Eliana or Gabe?
Hi, sorry about that. Can you hear me now?
Yes, we can. We can. Now it's good. Go ahead.
Okay, awesome. So we first looked at the albutrot and we prepped a silicon wafer with our sample. And we looked at it through atomic Morse microscope to look at it with the topographical features, and the picture is shown below. Then we looked at Raman on 2 separate samples. One was a 500 milligrams per milliliter hydrogel, and the other was powder. And the hydrogel ones are to the left, the top and bottom, and you can see how they're incredibly similar, indicating that the gel is nearly homogeneous, while the powder is the 2 on the right, which can be seen that they're greatly different when they focus on different microscopic areas, showing that the powder is heterogeneous. Lastly, we did toxicology testing, and we did it with 2 separate exposure times. One was a 1-hour exposure on 3 different kinds of cells, MDCK2, gingival keratinocytes, and HUBEC. And the first thing you can see is the EVOS microscope, showing that they're both similar, regardless if they were treated with the EPM or not, and there was no statistically significant difference. Then we did a 24-hour exposure version on human dermal fibroblasts. And again, you can see that the EPM had no adverse impact on the cell count of the cells that we use.
So for our future work, we hope to do mass spectrometry and NMR spectroscopy to further characterize the polysaccharide biopolymer and understand its properties. And we also hope to finish the antiviral testing and the rheology testing. In conclusion, we would like to acknowledge and thank all of our mentors throughout the program for all of their help and guidance. Thank you.
Hi everyone, I'm Rick, and with my partner Stephen, we'll be presenting about the properties and applications of biopolymers and nanocellulose. Today, we face a big problem with soil. As the effects of climate change become more and more apparent, the ability of soil to retain water and maintain structure has become vital to the reduction of soil erosion. We believe biopolymers and nanocellulose may be the answer to addressing these important issues. For this project, we used EPS biopolymer extracted from Rhizobium tropicae bacterium and nano-sized cellulose fibrils, also known as nanocellulose. These polymers have shown great potential in their ability to slow evaporation, bind water, and lower the freezing point, among other properties. Our objective is to determine how solutions with different concentrations of biopolymer, nanocellulose, and clay change our properties of interest, which are freezing point depression, water retention, percentage of non-freezable water, and the method of evaporation. Through this, we can compare the effectiveness of biopolymer versus nanocellulose and how these two polymers can be most effectively used in the natural soil. Now let's take a look at the results. At the top, we have the graphs for the contact angle measurements. A goniometer was used to take pictures of water droplets on pure silicon wafers to determine how these droplets evaporate. Our analysis of the images shown in the top left indicate that the biopolymer evaporates height-wise, meaning that the height of the droplet decreases faster than the width, creating a coffee ring type shape. The water in nanocellulose droplets evaporate differently. The height and the width decrease at the same rate, which keeps the droplets intact. We quantified our conclusions with the top right graph, showing the ratio of the width to height at different times.
All right, moving to the middle, we have the freezing point versus clay percentage found through DSC analysis. We found that the amount of freezing point depression increases with increasing concentrations of biopolymer, which follows the principles of freezing point depression. Nanocellulose has a lower freezing point at equal concentrations compared to biopolymers. Our microscale evaporation analysis shows that biopolymers has a lower evaporation rate than nanocellulose and water, having better water retention rates. Finally, moving on to the bottom left, our DSC results show that non-freezable water percentage versus concentration, indicating that the higher the biopolymer concentration is, the more non-freezable water is present, as more biopolymers can bind to the water. Furthermore, in the graphs with the non-freezable water percent versus clay percent, clay is shown to have little significant effect on the percentage of non-freezable water. However, it still shows that higher biopolymer concentrations generally has more non-freezable water, as previously stated. To conclude, biopolymer and nanocellulose solutions may be the answer to help prevent soil erosion, freezing issues with infrastructure, and other problems. In the future, we want to look further into the other properties that may affect how these solutions behave, such as pH and their viscosity. But from this study, we conclude that the efficacy of biopolymer and nanocellulose in solving the problems of soil and infrastructure is significant, and its use is possible and recommended. Finally, we would like to thank Dr. Rafalovich and Yiwei for their help and dedication to this project. Thank you.
In addition to the experimental...
Oh, I should say just an introduction to the next one, that theory and experiment are very closely linked, and it's one of the hallmarks of fruitful collaborations. Jerry.
In addition to the experimental studies conducted on the use of biopolymers as a soil additive, our group seeks to characterize how charge induces the network formation of biopolymers and clay fillers using coarse-grained molecular dynamics computer simulations. In each of our systems, we initialize 5 types of particles: the solvent, the backbone architecture polyelectrolytes, the rigid multi-bead fillers, and the respective counterons to the polymers and fillers. Next, we equilibrate our systems under constant NPT conditions with the finitely extensible nonlinear elastic potential to describe the polymer bonds, the soft potential, the Lennard-Jones potential to characterize dispersion forces, and the electrostatic potential to describe pairwise interactions between charged particles.
After equilibrium, we'll perform 3 tests under constant MET conditions: the stress autocorrelation function, the step strain test, and radial distribution function. Using the Green-Kubo relations, the stress autocorrelation function allows us to determine the system's viscosity by integrating from 0 to infinity with respect to time. If the integral diverges, then it indicates that like the infinite viscosity implies that like gelation has occurred. However, according to our results for both biopolymer charges, magnitudes of Q equals 1 and Q equals 2, the stress autocorrelation function converges to 0, which indicates that our system exhibits liquid-like behavior.
The second test we performed was a step strain test in order to measure the mechanical stability of our systems. During the test, each system was elongated by 20% and allowed to return to equilibrium. We expect to see convergence to a non-zero value of stress if the systems exhibit gel-like behavior. For both charge magnitudes Q equals 1 and Q equals 2, we see a decay of stress to a value of 0, supporting the conclusion that our systems do not undergo gelation. Finally, the radial distribution function plots the average number density of the polymers as a function of distance from each filler. All of our systems did not show significant peaks of large magnitudes around the cutoff distance of 1.2 sigma, indicating that polymer filler adsorption did not occur.
Our data supports the conclusion that both biopolymer charge magnitudes of Q equals 1 and Q equals 2 does not in fact induce gelation, nor does it lead to significant network formation. For our next steps, we first plan to run tests for polymer absorption as on a wall of charge while varying the charge of the polymers. This will allow us to see at what charge magnitude polymer filler absorption might occur. Next, we plan to run tests on more charge magnitude values in patterned or random charge distributions. Additionally, we seek to develop and implement new system parameters to characterize molecular geometries, physical moduli, and filler intercalation mechanisms. All in all, we would like to thank Dr. Rafalovich and the Garcia program for providing us with the opportunity to conduct this research. In particular, we also thank Joe McSaha and Dr. Gersape for their mentorship in this project. Thank you.
Any questions from the audience? Okay, then we will continue on to our next session. And again, a shout out to Dr. Steve Larson, who's also in the audience, for having been a wonderful mentor to the students and an inspiration to get these projects going. Thank you, Steve. Our next session, in silico and in vitro biology, guided by Professor Yufan Deng. And this is an ideal example of in silico trying to make biological systems sort of a twin in computational twin, and the ability to predict phenomena through computation. For example, they did a lot of work this year on COVID, and working with COVID is very difficult, but trying to model it is more achievable. So thank you very much, and please start the session.
Hello, today me and my partner Evan will be presenting on our project, a machine learning approach to molecular dynamics, predictions of SARS-CoV-2 spike protein denaturation at varying temperatures and pH levels. The COVID-19 pandemic is caused by the SARS-CoV-2 virus, and an important structural component of this virus is the spike protein, which initiates viral infection by binding to the human ACE2 receptor. This makes it vital to understand under what environmental conditions the spike protein denatures, since this will lead to deactivation of the virus and prevent it from infecting others. For this project, our specific aim was to examine under which temperatures and pH levels the spike protein will denature. One important way of studying structural changes that could tell us more about the denaturation of the spike protein is through molecular dynamic simulations. However, these simulations are very computationally expensive. And so because of this, our goal was to develop a machine learning model to predict the denaturation points of SARS-CoV-2 at different, of the SARS-CoV-2 spike protein at different temperatures and pH levels based on existing molecular dynamic simulation data.
First, we created a model for the root mean square deviation metric, which measures the change in position of each atom in the protein. We implemented 3 different machine learning algorithms, k-nearest neighbors, random forest, and a neural network, and found that the neural network performed the best as the only model with less error than the actual simulation. After this, we applied the neural network algorithm on the 2 other metrics, the main chain to main chain hydrogen bonds and the solvent accessible surface area, and also achieved errors smaller than that of the simulation. The last thing we did was visualize how these models would predict on a range of temperature and pH inputs with a heat map, as you can see on the slide. Moving forward, we're going to use the simulation to collect test data from these input ranges to understand how accurately the model predicts across these ranges. We'd like to give a big thank you to our 3rd group member, Ayush, who unfortunately couldn't make it today because he had school, and also to our mentors, Zuyuan, Professor Deng, and Professor Zhang. Thank you.
Okay, hello everyone. My name is Eric Chen, and today I'll be presenting my work on creating coarse grain models of solvated fibrinogen to expedite molecular dynamic simulations. So to understand it, we first have to understand what coarse graining is. Normally, in molecular dynamic simulations, which simulate the motion of proteins, each and every one of the millions of atoms within the proteins are simulated by the computer. So these simulations are naturally very computationally intensive, inefficient, and they can't really reach simulation times over a few milliseconds. Now, coarse graining greatly reduces the computational power required by reducing the number of particles that need to be simulated by the computer. It does so by essentially clustering together groups of atoms into singular particles and then accurately calculating both the structure of and the intermolecular forces between these particles, thereby allowing simulations at much larger time intervals. Now, my protein of interest is fibrinogen because of its critical role in thrombosis. A coarse grain model of fibrinogen already exists, but it's situated in a vacuum. Fibrinogen is naturally solvated, so we still need a coarse grain model of solvated fibrinogen. I've already created a coarse grain model of fibrinogen in a solution and an all-atomic model of solvated fibrinogen to use as a baseline. I've also created minimization simulations of these models that are currently running on a supercomputer and should be done in a few days. Then I intend to take measurements like the RMSD and RMSF, as well as create graphs like Ramachandran and Tika plots, which can compare the results of the all-atomic and coarse grain simulations. Then, based on the similarity, I'll know how effective the coarse grain model was. Finally, I would like to thank Ziji Zhang, Dr. Zhang, and Dr. Deng for their guidance and help. Now I'd also like to thank Dr. Rafaelovich and the entire Garcia program for facilitating this project. Thank you.
Hi, my name is Amit Saha. Today, I'll be presenting my project under Dr. Yuefan Deng, Accelerating Multiscale Blood Flow Modeling with Computational Fluid Dynamics. So cardiovascular disease, or CVD, is responsible for upwards of 17.9 million deaths per year. Many of these deaths occur because of thrombosis, otherwise the formation of blood clots. To better understand thrombosis, we have to model it. So multiscale modeling, or simulation at multiple time and length scales, can be used to simulate thrombosis. Previous literature indicates that the use of molecular dynamics and dissipated particle dynamics to simulation methods is accurate but slow. Dissipated particle dynamics specifically approximates fluids with particle flows and resolves detail that may not be necessary. This project examines the usability of computational fluid dynamics, CFD, as a much faster alternative to dissipated particle dynamics, which can be even further accelerated by orders of magnitude with neural networks. Furthermore, we aim to implement CFD with an adaptive, utilizing an AI adaptive time-stepping algorithm, or AIMTS. First, we aim to validate the use of neural networks for CFD. We trained a neural network with the Fourier neural operator architecture for fluid flow with an error rate of 1.6% and a speedup of 2.3 orders of magnitude relative to traditional CFD, as we can tell from the inference time figure in table 1. Secondly, we tested traditional CFD methods to determine the efficiency of CFD relative to DBD in existing simulation software or LAMPs. We first developed topology files and then implemented continuum methods in our simulation software, resulting in a dramatic speedup of approximately 9 times relative to DBD. Our future directions are to implement the neural network in the simulation software and to implement the AIMTS algorithm, allowing for the greatest computational efficiency and allowing us to use blood flow modeling for therapeutic applications. I'd like to thank Dr. Rafailovich and the rest of the Garcia staff for making this project possible. And I'd especially like to thank Yikong Zhu and Dr. Yuefan Deng for their constant support and guidance. Thank you all for listening to my talk.
Good morning, my name is Yuhe Wei, and I will be presenting my project on generalized Lena-Jones potential using physics-informed neural network. Platelet dynamics play an essential role in understanding the mechanisms of blood clotting, but the traditional approach of molecular dynamics simulations is very computational inefficient and time-consuming. My work aims to simplify the computation of Lena-Jones potentials, which is the intermolecular force fields between interacting platelets, through an analytic approach of discovering the prediction curve and learning parameters for the generalized equation. The quantile transformation converts the large datasets to a Gaussian distribution, thereby improving the accuracy of the prediction curve. We implemented the Earth function-based multivariate adaptive regression spline model to find the prediction curve of LJ potential, as shown in the figures below. Physics-informed neural network is also used to find the governing force field parameters and orientation functions represented by lambdas in equation 1. PINN embeds fundamental physics principles as regularization factors, constricting the range of solutions and improving the precision of the function approximation. We will assess the PINN training with a customized loss function to penalize the difference between predicted LJ potential and true LJ potential value. From the neural net framework, we will find the generalized equation by investigating the physics-guided equation. Lastly, I would like to thank all my mentors, Jason, Yitong, Professor Zhang, Professor Deng, who made this research possible. Thank you.
Hello, everyone. My name is Matthew, and the people here presenting with me today are Agam and Lian. And for our research, we studied the mechanisms of virus-induced thrombogenicity. So for some background information, fibrinogen is a plasma-soluble glycoprotein responsible for thrombogenesis, the formation of blood clots. While thrombogenesis is an important process, excessive blood clots can cause serious health conditions like strokes and heart attacks. Previous literature shows that fibrinogen polymerizes in virus-infected cell cultures, but the reason why is unclear. The purpose of this project is to investigate the connection between virus-induced lipid secretion and fibrinogen fiber formation.
To study the formation of fibrinogen fibers on surfaces, solutions of polybutadiene, polylactic acid, and polystyrene were spun cast onto silicone wafers for their thrombogenic and biocompatible nature. Fibrinogen was added to the films, imaged through AFM, and analyzed for their physical properties. The deflection images reveal that fibrinogen is indeed able to form fibers on flat hydrophobic surfaces.
Moving on to the second part of our project. So in the human body, hydrophobic substances are secreted by virus-infected cells. To visualize this, dog kidney or MDCK2 cells and human endothelial cells or HUVEC cells were plated and infected with H1N1. Human gingival keratinocytes as models of epithelial cells were also infected with H1N1 through direct infection and conditional media taken from previous MDCK2 cell cultures. This specific virus was used because it provides insight into COVID symptoms. After fixing and drying, cultures were stained for actin, lipids, and DNA, followed by EVOS microscopy analysis. MDCK2 cells infected with H1N1 secreted lipid droplets, while both the control and direct infection HUVEC cultures did not display lipid droplet formation. However, HUVEC treated with the conditional media taken from infected MDCK2 cultures developed lipid showers, demonstrating that the hydrophobic secretions of an infected culture spreads to the surrounding tissue. The same results were observed with human gingival models.
For future steps, P12 and anti-clotting peptide will be tested to reduce fibrinogen aggregation. We would like to thank our mentors, Conche Feng and Adam Hansen, as well as Dr. Rafailovich for guiding us through this research. Thank you.
Hello, everyone. My name is Michael, and my partner is Rami, and our project is on the in silico modeling of the ideal binding sites for polylactic acid chains and fibrinogen. So the purpose of the overall project is to create a non-thrombogenic material for implants. Medical implants that come into contact with blood will interact with fibrinogen, so it is important to determine how fibrinogen will bind to materials such as polylactic acid. Our project focuses on creating a model that would better simulate the interactions between fibrinogen and various materials for studying purposes.
So fibrinogen is made up of 3 domains, 2 D domains on both ends and an E domain in the center. Using Autodoc Vino, we located potential binding sites for the 2 PLA polymers, lines 3 and 5, for each of the 3 domains. Out of these potential options, we defined the optimal binding sites per domain using the lowest docking score, indicating the highest binding affinity. We then superimpose individual domains of the 2 optimally bound PLA polymers onto a completed fibrinogen structure. These results can be extrapolated to show that if placed on a PLA or polylactic acid surface, fibrinogen would bind to the optimal points shown in figure 4 in red. And figure 5 is not drawn to scale, but it is a diagram of what this positioning would look like. Comparing the 6 optimal binding sites, we can surmise that the docking scores for PLA length 5 were less than those for length 3. And for PLA length 5, both the D domains had similar scores, while both the D domains had similar scores, while for PLA length 3, the E domain and the right D domain had identical scores, of the D domain on the left was greater.
We would like to thank our mentor, Bernard Essuman, Professor Deng, and Professor Zhang. Thank you all for listening.
Hey everyone, my name is Pratham Patel, and my project is about the optimization of the Lern-Jeffrey orbits equation for plate rotation. So nowadays, molecular dynamics simulations are very computationally intense. Specifically, it takes around 54 hours to simulate one full plate of rotation. And so the research specifically pinpoints on decreasing computational intensity of plate rotation by implementing an improved Jeffrey orbits equation and integrating it into an RNN to predict plate rotation. Specifically, this improved Jeffrey orbits equation has new dynamic parameters, including fluid interaction, oblate deviation, and deforming capability. These dynamic parameters help increase the accuracy of the model as a whole. And so currently what we've been doing is we've been applying wavelet transformation and a moving average filter to the data in order to denoise it. And we have a large amount of ground truth MSM data, which we'll be using it to train our biomodel. Specifically, I'm using PLA 139 in order to train our biomodel. Finally, we'll be implementing this new ground truth MSM data to generate platelet movement plots, including rotational velocities and velocities in the X, Y, and Z direction. Finally, I'd like to thank my professor Yufan Deng, my graduate student Yukong Zhu, and Dr. Peng Zhang as well. Thank you.
Hi, everyone. My name is Ishan Singhal, and my project is using recurrent neural networks to stimulate coarse-grained proteins in silica. Molecular dynamics uses fundamental physics principles to calculate the forces on each atom, allowing experimentation without the need for laboratory procedure. However, it requires heavy computational power and months of time. Thus, coarse-graining algorithms have been developed which attempt to derive these forces in a more efficient manner, allowing for longer simulations and more protein observation with a limited cost of accuracy. Currently, the CGNet approach employs simplistic neural networks with features calculated from protein model atoms to predict the forces. In order to improve the accuracy and speed of the program, we propose a novel architecture using recurrent neural network layers, which specialize in time-step data. Accuracy of each approach was determined using RMSD of the trajectory and KL divergence in MSE, which were obtained from the Ramachandran plots for the alanine dipeptide data as shown. However, Tika energy plots were used for signaling data due to the greater structural complexity. Preliminary testing with 1,000 frame alanine dipeptide data demonstrates higher model accuracy with the RNN model. However, we are currently running 100,000 frame signaling data on the IBM supercomputer for final results metrics. For this project, I would very much like to thank my grad student, Ziji Zhang, as well as Dr. Zhang and Dr. Deng for allowing me to participate in their research lab. Thank you.
So individuals with medical implants are at an increased risk for developing blood clots, which can lead to life-threatening outcomes. Platelets aggregate to form these clots and with the use of fibrinogen, as shown in the diagram. In order to inhibit this process, a molecule would have to bind to the fibrinogen alpha C domain, and one such candidate is P12. So our methods were over the past 3 days. The above experiment was conducted. The first column is the controls, and the rest are various applications of P12 and fibrinogen in different orders. So the 10 microgram per milliliter creates a thin film of fibrinogen onto the surface. The P12 is a substance that effect is being tested, and the 4 milligrams per milliliter of additional fibrinogen is supposed to bind with the film that already forms if it exists. So our results suggest that adding P12 to fibrinogen does decrease fibrinogen formation. Fibers that do form are thin and much less interconnected than the controls. The most effective implementation was adding P12 a day after the 10 microgram per milliliter fibrinogen was plated. And I would like to thank Jessica and Dr. Rafailovich so much for their support, especially with all of, as you guys saw, the how this was sort of last minute, but they were willing to assist me despite and allow me to stay up till 3 in order to complete the slides.
Thank you. Thank you very much. And I'd like to introduce Professor Yufan Dang, who really mentored this extensive symposia. Yufan.
Well, it's a great pleasure. Thank you, Professor Rafailovich, for sharing the Garcia talents with our group. And many projects we wish to tackle have been enabled by the Garcia scholars, as you can see from their 6 or 7 presentations. I'm very impressed and almost speechless. Good luck to everyone, including students from our group. And we'll welcome you back and continue working on those projects. And hopefully we'll write a high quality papers. And also I will go to great colleges to continue doing science you like. Thank you, and good luck with the symposium.
Thank you very much. And if there are no more questions, then I'd like to introduce the next symposium, which is renewable energy generation. And here we're going to talk about different kinds of cells. These are the cells that have electrodes coming out of them and generate energy. And as I said, the Garcia program does both materials and biological materials. To introduce the session, we have Yifan Yin and Haoyan Fang, 2 graduate students in the Garcia program.
Hello, everyone. My name is Haoyan Fang. Can you hear me?
Yes. Yes, we can.
Okay. In this section, we have mainly 2 different directions. One is fuel cell and one is solar cell. Renewable energy is very important in our daily life. And the main problem for fuel cell is the slow oxygen radiation and the high cost. And for fuel cell part, we have 2 projects. One is cellulose to solve the problem of the high cost of catalyst on fuel cell. And another is the oxygen reduction. We have a project which is silver nanoparticle catalyst to solve this problem. And for solar cell, the topic is like use 2D perovskite to improve the efficiency and the durability of solar cell. Yeah, and let's move to the students about energy, give their presentation.
Hello, everyone. We, the Fuel Cell Group, have 2 projects to present to you this year, and the first is using acid-treated cellulose fiber membranes to enhance the performance of proton exchange membrane fuel cells.
So a bit about proton exchange membrane fuel cells. At the anode, hydrogen gas is supplied and it produces protons and electrons. The protons flow through an external circuit that can produce power. And then the electrons flow through the external circuit and the protons move through the membrane. Then at the cathode, the protons combine with the electrons and oxygen gas to form water.
Migration of hydronium ions across the membrane is a process that's facilitated by the expensive Nafion membrane, which research has been exploring cheaper alternatives to, like cellulose, as it's a naturally occurring polymer. And it's even been shown to show enhanced power output when treated with RDP, a flame retardant polymer.
Our goal for this project is mainly to improve the power output obtained from fuel cells using a cellulose-based electrolyte membrane treated with additional variables such as different acids or the alteration of the fuel cell parameters. An older Armstrong cellulose paper from 2019, along with a newer Armstrong cellulose paper from 2022, were given 4 different treatments ranging from a control with only the flame retardant RDP to RDP combined with 1 molarity citric acid, 1 molarity phosphoric acid, and a mixture of the previous 2 solutions. Additionally, recent explorations in the electrolyte membrane have led to more modifications in the fuel cell parameters, such as the use of pure oxygen supplied to the cathode compared to air, which resulted in significantly higher power output.
Our results show that acid treatment was effective on all types of cellulose paper we tested and produced a power output of double that of control group. Additionally, we found that mixing citric and phosphoric acid can improve performance at higher temperatures. We did SEM analysis, which shows that there is no significant difference between the chemical compositions of the different cellulose papers. And finally, through EDS analysis, we found phosphorus, which indicates that RDP was successfully infused into cellulose paper.
Further research of RDP-treated cellulose filter paper as a viable alternative to Nafion for proton membrane exchange remains necessary. As for future work, we are considering a variety of different next steps, such as testing nitrocellulose on the fuel cell testing station, incorporating graphene oxide into the cellulose filter paper samples, and using a pure oxygen supply to the cathode. Additionally, continued investigation into different combinations of acids and molar concentrations is currently underway to enhance their potential effects on the PEMFC power output. Thank you. So now I would like to present our second project, investigating the silver nanoparticle catalysis mechanism in alkaline anion exchange membrane fuel cells.
So anion exchange membrane fuel cells are fuel cells with an anion exchange membrane that facilitates the movement of hydroxide ions from the cathode to the anode, and in order to produce water.
AEMFCs are a type of hydrogen fuel cells, which all produce power with the only byproduct being water. AEMFCs function in saltwater and basic environments, unlike PEMFCs. Recent studies prove that AEMFCs, however, have lower durability than PEMFCs because the platinum catalyst penetrates into the membrane. Recent study also shows that the power output is enhanced if platinum catalyst is coupled with silver nanoparticles that are sprayed onto the membrane.
Our objective is to better understand the silver nanoparticle catalysis mechanism by studying the effect of when we deposit a monolayer of it through an LBT at different surface pressure versus when we spray it directly onto the membranes and electrodes of the AMFC. We also wish to perform a durability test to see if the silver nanoparticles provide any special enhancement on the AMFCs there as well.
Now for the results. We've tested 5 nanoparticle deposition scenarios so far, including a control, 5 and 10 millinewton per meter monolayer depositions, a membrane spray, and an electrode spray. As you can see in the bottom center, in terms of maximum power density, the 5 millionewtons per meter surface pressure fuel cell performed the worst, followed by the control, the 10 millionewtons per meter, the sprayed electrodes, and finally the sprayed membrane, which performed the best. The 10 millionewtons per meter deposition and the 2 sprays contained the same number of nanoparticles, but the membrane spray had a significantly higher power density, indicating a mechanism that does not benefit from the change in nanoparticle shape due to LBT deposition, but does involve catalysis of a nanoparticle membrane interface.
The TEM images and size distribution graphs we generated of silver nanoparticles shows that the monolayer deposited at 5 millionewtons per meter surface pressure has a higher size distribution, and some of the nanoparticles appear to be platelet shaped. The monolayer deposit at 10 millionewtons per meter surface pressure is more uniform with the lower size distribution, which may be the reason for producing a higher power density, but that could also be attributed to the fact that there were simply more nanoparticles at the 10 millionewtons per meter surface pressure deposited monolayer.
For future testing, we will be performing durability tests to see if the silver nanoparticles block platinum from permeating into the membrane. We would like to thank the Garcia Center for providing the resources necessary to conduct this research, along with the many individuals who helped facilitate this research, including our graduate students, Aniket Raut, Hao-Yan Fang, and Dr. Farabi Rahman, along with Ms. Israf and Dr. Rafalovich.
Hello everyone, today we will be presenting our project Surface Passivation using 2D perovskites toward efficient and stable perovskite solar cells. So our goal is to investigate the effects of applying 2D perovskites as passivation layer on top of a 3D active layer. As a result, we constructed a 2D 3D heterostructure by reacting BI with the surface of the 3D perovskite layer. And we dissolve BI of different concentrations in IPH, the influence of the thickness of the 2D perovskite layer on power conversion efficiency. And we investigated the surface morphology, crystal structure, chemical interaction, and PV performance of our device.
For the fabrication process, we first made a substrate composed by FTO glass, compact, and mesoporous titanium dioxide. Then we used solvent engineering to construct a 3D perovskite. During the process, we did anti-solvent dripping using chlorobenzene, followed by annealing. Next, we spin casted 2D ligand onto the 3D perovskite to react and form a 2D perovskite, followed by annealing as well. Our SEM samples indicated that while pristine samples had defective boundaries, passivated samples had more fully covered grains, with the 3BAI sample yielding the optimal results. Our XRD analysis indicated that BAI coating didn't change the FAPI crystal structure.
Our XPS test showed that at the top surface region of the perovskite film, which was around 5 nanometers, the peak corresponding to lead ions in the 3 passivated samples shifted to lower binding energies compared with the pristine sample, indicating that 2D passivation made the whole system more stable. Finally, our PV curves indicated that the 3BAI sample displayed the lowest hysteresis and the highest power conversion efficiency and open current voltage among the 4 samples. In conclusion, the 3BAI sample yielded optimal SCM results and also exhibited high PCE with negligible hysteresis. 2D coating also didn't significantly alter the 3D crystal structure. Lastly, we would like to thank our mentor, Yifan Ying, Professor Rafalovich, and of course, the Garcia program. Thank you.
Thank you. And I should also mention before the next presentation that this group participated entirely on Zoom from China with a 12-hour time scale. So instead of these people started every night at 10 p.m. after a full day of other activities. So thank you so much.
Hello, everyone. I'm Tony, and I, along with my research partner Ming, will be presenting our research on modeling dendrite growth in alkaline metal batteries. So while lithium batteries are more widespread, sodium batteries are significantly cheaper and environmental friendly. However, both face a common problem. Dendrites, which are protrusions on the anode surface, decrease battery performance and lifespan while posing safety concerns. Methods of reducing dendrite growth for lithium are already proposed, but our goal is to determine if they are viable for sodium.
We first compared 2 forms of charging methods, CCCV and pulse, on their charging effectiveness and their limitation of dendrite growth in lithium batteries. CCCV charging is shown in the top left graph, charging at a constant current and then switching to a constant voltage, while pulse charging is shown in the bottom left graph, giving the battery regular breaks to absorb the lithium ions. We simulated a lithium half cell using lattice-Boltzmann methods in the Palabos library in C++, and with the Stony Brook Seawolf supercomputer, then simulated charging that battery using both methods.
Our qualitative analysis of dendrite formation was done using ParaView, and quantitative data was processed using Python. As seen in the images to the right, charging the battery led to visible growth of dendrite structures. However, pulse charging at a frequency of 9 seconds increased charge time from 348 to 408 seconds, yet lowered the RMS size of dendrites from 3.1 to 2.2 micrometers and reduced their maximum heights from 45 to 42 micrometers. This shows that pulse charging, while taking longer, does lead to reduced dendrite growth, vastly improving the safety and lifespans of lithium batteries.
Despite their advantages, one major hurdle for the adoption of sodium-ion batteries is that dendrites grow and form on them faster than on lithium. We conducted a literature dive and began modifying the lithium-ion model to instead simulate a sodium-ion half cell while simultaneously optimizing the parameters. By running the sodium-ion model, we hope to gain novel insight on the formation of dendrites in sodium-ion batteries and how different charging methods may prevent it.
Overall, our research confirms that pulse charging improves reliability and safety in lithium-ion batteries, and we aim to investigate if it can do the same in sodium-ion batteries. To conclude, we would like to thank our grad student Jolyn, Dr. Korsape, Dr. Rafailovich, and of course the Garcia program for their mentorship and support throughout the project. Thank you.
Thank you very much. Questions? Okay, please shout out or put them on chat if you do. You can always reach us later if you have questions after this session. And our next presentation is, we emphasize a lot to our students that reaching out to the community in communication and teaching is very important. And we'd like them to be able to give as much as to take from this program. And these students prepared a wonderful video as an asset and as a resource for teachers teaching basic science. So please present your video. Tangri.
Have you ever wondered what would happen if you touched a freezing cold drop of liquid nitrogen? Or why water hovers instead of evaporating on a hot pan? The answer is the Leidenfrost effect. Today we'll be talking about the chemistry behind the Leidenfrost effect and how you can demonstrate it yourself at home. Let's get started. So all we need is a pan, a stovetop, and something like this to drop small amounts of water. Now let's get this heated up. It'll take a bit. Don't forget to wear something to protect your hands. I'm using oven mitts, and always use adult supervision.
A few moments later.
Alright, it looks about ready. Let's see what happens when I drop some water on the pan. Now let's try the other side.
Why do the water droplets levitate? When a drop is put on a really hot surface, the bottom part vaporizes so quickly that it creates vapor gas between the hot surface and the rest of the droplet, allowing the droplet to glide effortlessly around the pan. The vapor gas also helps the water droplets roll by itself on the other side of the pan. Each time hot vapor is produced, it acts like a small jetpack, pushing downwards and causing the droplets to roll. The vapor conducts heat slower than the regular liquid, which means that it takes up more time for the hot surface to vaporize the rest of the droplet. This helps insulate whatever is behind the liquid. This vapor cushion stays until there's no more liquid left, so the drop will continue to levitate until it disappears. You can see this happen whether the liquid is really hot or really cold. Does this mean that we can swim in a pool of molten lava? Definitely not. But does this mean that scientists can wow their families and friends with levitating water? Yes, it does. Thank you everyone for watching.
And thank you, Rick, Xian, and Stephen Yang for this wonderful and entertaining production.
Have you ever wondered?
Yep. Thank you very much. We'll have short clips as interspersed between the sessions to show you the talents, the multiple talents of our students in the Garcia program. The next session, neuro and osteodifferentiation of dental pulp stem cells, and this will be introduced by Anna Cho, our undergraduate guide.
Hello, everyone. Welcome to session 4, Neuro and Osteodifferentiation of Dental Pulp Stem Cells. So during the summer, the students have studied about the effects of different factors such as magnets and clay on the rate of neurodifferentiation and osteodifferentiation of these dental pulp stem cells. So today, we have evaluating the effect of increased resorcinol diphenyl phosphate concentration in clay on the neuronic differentiation of dental pulp stem cells by Hilal Shine and Rebecca Sullivan. And we have investigating the effect of static magnetic fields on the differentiation of these dental pulp stem cells into neurons by Oran Goodman and Nikita Srinivas. Investigating the effect of a dynamic magnetic field on the rate of dental pulp stem cell neurogenic differentiation by Alexander Wang and Sahil Sood, and the effect of microsphere growth on the static magnetic field on osteogenic differentiation of dental pulp stem cells by Angeline Sun. Please enjoy listening to this session. Thank you.
Hi, everyone. We'll be presenting an analysis on the effect of increased resorcinol diphenylphosphate concentrations in clay on the neurogenic differentiation of human dental pulp stem cells. Some background information is that currently there are many neurogenic diseases which we have no cure for. Our goal was that through using human dental pulp stem cells, we could differentiate those stem cells into neurons and thus create cures for those diseases. Our hypothesis is that increased concentrations of RDP will result in a greater and more effective rate of differentiation into neurons.
Our materials include pure PLA, TCP, 20% RDP clay, 30% RDP clay, and 20% 30B clay, which were coated onto silicon wafers as surfaces for stem cell differentiation. Moduli and EVOS imaging were performed on day 5 after plating and day 9 after changing neural media, and we were performing further modulus and EVOS on day 28 after changing neural media, as well as an RT-PCR test to ensure that the cells did in fact differentiate into neurons. The top chart shows the results of the day 5 and 9 moduli, which reflect cell stiffness. As you can see, the cell stiffness decreased between day 5 and day 9, which is a common trend among stem cell differentiation. The bottom chart shows the rigidity of the films.
We also compared surface roughness of the films compared to pure PLA. We noted that between 20% clay and pure PLA, there was an increase of surface roughness of about 92%. Between 20% RDP and pure PLA, there was an increase of surface roughness by about 89%. And between 30% pure RDP and pure PLA, there was a surface roughness increase of about 91.64%.
The day 5 EVOS images are shown above at 20 times zoom and 10 times zoom. The blue represents the nuclei, which were stained using DAPI staining, and the red represents the cells, which have a red fluorescent protein.
Additionally, we did day 9 EVOS imaging, and we noted that compared to day 5 EVOS imaging, that there appeared to be what seems like ganglion structures, which would be evident of neuronal differentiation. So this will then be confirmed using day 28 RT-PCR. Thank you.
Hi, my name is Nikita and my partner is Iran, and we're going to be presenting our work on the effect of static magnetic fields on the differentiation of dental pulp stem cells into neurons. So neurons are cells that make up your brain and nervous system, and they are essential for proper communication and control in the body. But when some trauma destroys these cells, adult humans do not have a way to regenerate them. So any way to create more neurons would be very beneficial in neurodegenerative medicine. Coming into this project, we believe that stem cells grown in the presence of a static magnetic field would differentiate into nerve cells better. And in this experiment, we tested this hypothesis using both plain PLA and a combination of this PLA with 20% RDP clay, which was shown to help differentiation in prior studies. The samples were switched from a stem cell media to a neuronal media after 3 days, and we used modulus testing and EVOS microscopy both before and after the media change to determine and compare rigidity data, as well as view the physical appearance of the cells as they differentiated.
So the day 5 and day 9 EVOS for the PLA inside the field seem to be differentiating very well. But interestingly, after day 9, our samples grown on the spun cast silicon started dying. This was weird, but in the end, we think that the electric charge produced by the semiconductive silicon under the magnetic field was the thing that killed the cells on top of it. And then what was interesting about the RDP samples in the magnetic field was that the films that we were growing the cells on started falling off, which in turn made the cells die. But we might attribute this to the ions in the RDP clay being moved around too much by the magnetic fields, because ions are moved in magnetic fields, thus weakening the film and making it fall off. And then finally, in figure 2, by day 9, the cells in the PLA in magnetic fields had all moved to the edges, which is weird because if something was going to kill all the cells, it would kill cells on the entire wafer, not just the middle. But what we think happened was that they moved to the edges where the silicon is actually a little bit rougher, and the cells can attach better due to this roughness. To combat all these causes of cell death, we are performing the experiment again on molded samples. So these molds are heat treated just not on silicon. So therefore they don't have the electric charge, and they're stronger. They don't have films that can fall off. So the RDP surface should not degenerate and kill the cells. Thank you so much.
All right, my name is Sahil Sood, and my partner Alex and I will be talking about the use of a dynamic magnetic field to stimulate differentiation. Now, similar to the past two projects, neurodegenerative diseases and CNS axon lesions cause major health problems because when neurons are degenerated, it's hard to replace them because of the intrinsic non-dividing and non-regenerative nature of neurons. So it's imperative to develop a protocol that can catalyze neurogenesis efficiently and in a manner that makes them easy to replace in patients that have long-term neuronal damage. So the reason we chose dental pulp stem cells particularly is because these are easily accessible multipotent stem cells that have a high proclivity to differentiate into several cell lines. And we wanted to control that differentiation from any specific cell line into neurons using neuromedia, nanoparticles, and a dynamic magnetic field. So the objective of this project was essentially to determine whether or not inserting iron oxide nanoparticles into DPSCs and stimulating them with the dynamic magnetic field would help increase differentiation. Now, for our methods, we accomplished this by spin casting 24 silicon wafers with either pure PLA or PLA with 20% RDP at 2,500 RPM for 30 seconds. That helped us create thin polymer films, and pure PLA was just our control. And then we plated the DPSCs onto the surfaces using an initial seating density of 1.5 times 10 to the negative, or 10 to the 5. And after, some of the samples received nanoparticles and some of them didn't. After day 4, each Petri dish that had nanoparticles was placed in a dynamic magnetic field. Each sample was exposed to the same conditions with 30 minutes of the dynamic magnetic field every other day for 2 weeks. And we then compared the cells with dynamic magnetic field treatment to the cells without dynamic magnetic field treatment. And I'll pass it over to Alex to discuss some of the results.
Yeah, so we conducted multiple testing for data collection throughout this experiment. The first one we did was on day 3 before neuronal media change from the dental pulp stem cell media. And we basically did modulus testing where we tested the rigidity of the cells in 3 different spots and on 3 different cells on each plate of the controls. So that would be on the TCP plates and also the PLA plates before any sort of magnetic field or nanoparticle treatment. And what we were expecting on these controls is for our day 6 modulus following neuronal media switch, we were expecting much lower moduli results because neurons are supposed to be much softer than the dental pulp stem cells. However, what we also saw on day 6 following neuronal media change was that the ones treated with nanoparticles and the dynamic magnetic field were much more rigid than the ones not treated with the magnetic field. And we believe that this is the case because the nanoparticles have attached onto the cell membranes, and therefore, since they're made out of iron oxide and gold shells, they're much more rougher and much more solid than the typical cell membrane. We also performed 2 EVOS testings throughout this process, one on day 3 before the neuronal media change and on day 9 after. And what we see on day 9 is on the exact same sort of surface, polylactic acid, there's a high morphology similarity to native neurons. So you could see on the photo on the right compared to the one on the left, there's like tubular development of neurons, axonal development, and eventually we're hoping to see some dendritic development as well to show that we have the morphology of a native liver. And then on day 28, we will perform modulus, EVOS, and RT-PCR to find out if we have the genetic traits associated with a native neuron. We'd like to give our thanks to Dr. Rafaelovich, Quan-Che Feng, Adam Hansen, Elias Georges, and Anna Cho for leading us through this experiment. Thank you.
Hi, I'm Angeline Sun, and today I'll be talking about our project on investigating the effect of microsphere formation and static magnetic fields on osteogenic differentiation of DPSCs. So first, on the microspheres, we are basically culturing cells to force them to aggregate and interact with each other in a 3D matrix. And this cell-to-cell interaction enhances extracellular matrix secretion, which will enhance bone regeneration. We cultured the cells in two types of wells, the first one being the suspension and the second being the agarose wells with different volumes, which are 50, 100, and 150 microliters of agarose. From the microscope and EVOS images on the left, we can see the agarose wells are better at forming compact microspheres than the suspension wells, and we further confirmed that through the Almar Blue assay because they showed higher fluorescence than the suspension wells. And next Monday, we will be performing RT-PCR to evaluate osteogenic gene expression. As for the static magnetic field treatment, we are using 3 types of scaffolds made of PLA, RDP clay, and iron oxide, each of which with different biological properties. And the cells on these scaffolds will grow under the absence or presence of magnets. So we hypothesized that since iron oxide is the roughest because it has the highest root mean square roughness according to the AFM measurements, its mechanical and magnetic properties will enhance osteogenic differentiation. Currently, the cells are all growing successfully on the 3 types of scaffolds as seen in the EVOS images, and we will use PCR to quantify gene expression, EVOS to image cell morphology, and SEM and Raman to analyze biomineralization. Lastly, I'd like to thank our mentor Quanche, the RUs, and Dr. Rafaelovich for making this research possible.
Thank you. Questions? And if there are no questions, our next session is yet again something different and something fascinating, and again inspired by Dr. Ellison. And these students work together with Professor Chakrabarty from the Department of Mechanical Engineering. Please take it away.
Hi, everyone. Through our project, we created a pipeline that would allow an operator to demonstrate a task, such as opening a door or moving an object in virtual reality. And by having a robot learn that task, we can have the robot further execute and generalize different motions with slight physical differences. This is called learning from demonstration, or LFD, but it comes with a caveat. The person's almost always physically there, which is inefficient and at times impossible. So in a future where we integrate robots in daily life, we need quick and efficient ways to teach robots, which is why we have created a VR pipeline.
So our goal is to create a VR LFD pipeline, but what exactly does that mean? Well, the pipeline that we've developed consists of 3 main parts: task constraint extraction, task generalization, and motion planning. For task constraint extraction, we created a virtual environment in Unreal Engine 4, added all the necessary objects that we might need, and we tracked the movements of those objects during the demonstrations that we performed in VR. We would then take the data, convert it into matrices, and move on to task generalization. Now, task generalization is important because it's based on the idea that reality is a bit more unplanned than virtual reality. So if in virtual reality, for example, you want to pour a liquid from one cup to another, in virtual reality, those cups will be a fixed distance from one another. But in reality, those cups could be farther apart, closer together, in all different positions and orientations. So task generalization is really what accounts for that unpredictability and allows us to guide our robot in performing manipulation tasks in a much more generalized sense.
The demonstration processing algorithm then outputs a reformatted trajectory for the robot arm's end effector, basically to a human arm's hand. Then this output is fed to a motion planning algorithm, which plans the trajectories of the 7 joints of the robot arm. Finally, the task is actually executed, and all that is just a high-level overview of the pipeline that we've made. During the remote part of the program, we then implemented the pipeline on our own computers, as opposed to desktops in the lab, showing the adaptability of our programs. We also streamlined the pipeline by automating things such as sending data between the computers and moving the robot arm to its starting position virtually. Further refinement is required, but we have a working pipeline at the moment. Thank you to Professor Chakraborty and our PhD student, Dashradan at the robotics lab at Stony Brook University for all the help and guidance. Thanks to the Garcia program and Dr. Rafaelovich for the support over the past few weeks. And thank you to all for listening.
Hello, my name is Benjamin Wen, and I'm here today with my lab mate, Edward Sun. We've been working with grad student Aditya Papinkar and Professor Chakraborty to build an end-to-end convolutional neural network guided point cloud segmentation system. For individuals living with diseases such as arthritis or cerebral palsy, a robotic arm has the potential to significantly increase independence. However, programming a robotic arm to be capable of interacting with the real world on its own comes with many challenges. One of which is, how does a robotic arm determine the 3D contour of an object? To address this issue, we've created a system that takes a 2D RGB image and a 3D depth image using the Intel RealSense D415. Then, on the 2D image, we apply the YOLO V5 neural network, which detects the object and applies a bounding box. On the bounded image, we deproject the pixels back into 3D values to get the segmented point cloud. I'll now pass it on to my lab mate, Edward, to discuss the rest of our methodology.
After we've localized the box image, we transform the box by applying rotation transformation and adding a translation vector. This transforms the box from the reference frame of the camera to the reference frame of the world. We then remove points based on the points that maximize in the world frame Z direction. Then we utilize a dbScan clustering algorithm to separate the point cluster sections by density and filter for the section that has the max number of points, which would always be the object itself. Lastly, we bounded the noisy point cloud data with a bounding volume to more accurately represent the orientation and size of the box. The results can be seen on the right. The blue box is right after the cropping, and the rainbow colored one is bounded and transformed box. And the last image shows the final results within the world frame, where the world frame and the camera frame definitions can be seen on the top left corner. We would like to thank our grad students, Aditya and Dr. Chakraborty, for allowing us to use the resources in their lab. We also like to thank Dr. Fralovich and the Garcia program for facilitating this project. Thank you.
Hi, I'm Alex Amadi.
And I'm Sergio Rosa.
Our project is a cross-platform transfer. So what is the main objective of our project? So our goal is to improve the quality of life for those who suffer from quadriplegia. Someone who suffers from quadriplegia would be paralyzed in all 4 of their limbs and would typically utilize an electric wheelchair. With this being said, they would typically, people that suffer with this disability wouldn't be able to move their neck, so their range of vision is somewhat limited. But our group sought out to find a solution to this.
Figure 1 is an image of a tongue-touch keypad. A tongue-touch keypad is a small keypad that is attached to a retainer with 9 sensor buttons that can act like a mouse for a computer.
So as Sergio mentioned, as buttons on the TTK is pressed, it would send out mouse inputs, and this would go to the Raspberry Pi. Once the Raspberry Pi receives it, it would send it to the Arduino through a serial port. Once the Raspberry Pi detects these, it would instruct the servo which way to move, and the camera system would be mounted on top of the servo. So in sum, as the TTK buttons are pressed, it would the camera would move accordingly.
Figure 3 is a picture of 3 wires soldered together. Originally, it would be 1 wire connecting the Arduino to a servo, but now the 3rd wire is necessary to connect to a 2nd servo.
So as you can see in figure 3 with the blue wire, that would originally be connected and it would go from the Arduino to only 1 servo, but we want to add a 2nd servo so we can mount a 2nd camera. So we would add the red wire that would lead to the 2nd servo. This would allow there to be 2 powers with 1 instruction. So then once all this is finalized, we want to make everything more compact. So we would put a box that had Raspberry Pi, Arduino, and then servo going out with the camera system mounted on top of that. Once that is all compact and everything, we would want to mount a small monitor on the armrest to show what the camera sees, and 2 buttons to move the camera left and right. Once that's mounted onto the armrest, there would be wires going to the back of the wheelchair where there would be a small box that would have all the things I mentioned before, and figure 4 would be used to clamp that to the back of the wheelchair. Now on to the future development.
Adding a 2nd servo motor will allow for a full range of view, with 1 servo going up and down and another going left and right.
So we're currently working on that right now, as I mentioned from figure 3. But we're also working on the next future development, which is making the TTK Bluetooth compatible. One of the biggest issues with the TTK is that the receiver connects with a serial port, which is very outdated today. So making it Bluetooth compatible would allow it to be utilized with a lot more other platforms or devices. This leads me to the next future development, which would be to allow the TTK to be applied to the wheelchair movement. If it's Bluetooth through some software or maybe connecting to the wheelchair directly, the TTK may be able to move the wheelchair with the corresponding buttons. With that being said, I want to give a big thanks to Dr. Rafailovich, Dr. Singh, and Mr. Hugh Rochert. Thank you.
Hi, my name is Yubo, and along with Eric, Junie, Judah, and David, we worked on emotion detection using deep learning in conjunction with digital image speckle correlation. So detecting micromotions of the face muscles has a bunch of practical applications. It can be used to identify muscle paralysis, distinguish people from each other, gauge reaction times, detect deepfake videos, and measure people's emotions. And digital image speckle correlation, or DISC, which was first developed for materials scientists to better understand mechanical stress, is a promising tool for muscle movement detection because it can detect extremely small movements in the face. And it works by comparing pairs of frames and generating a vector map that tracks the motions of key points between the frames. So previous work using discs for facial analysis depended on the camera and subject being completely stationary, and we utilized a different method to get around this issue.
Yeah, so in order to run DISC, we first detect the face in each frame of the video using the open source media pipe library developed by Google, and create canonical faces with the same size and position for each frame to mitigate head movements in the video, as you can see in the figure from step 1 to step 2. And these resulting canonical faces are then used to run DISC, which uses optical flow methods for computation. And the resulting vector field is plotted onto the canonical images, as you can see in the third step, and then inverse mapped using affine mapping, converting the canonical faces back to the original video frame, as you can see from the third step to the final step. Ultimately, this process produces a video analogous to the original input, but with an overlay of the vector map tracking the micro movements of the face.
We are now trying to use the CK Plus dataset, which contains hundreds of different videos already labeled with various emotions. And we are trying to utilize and optimize a currently existing convolutional neural network, an architecture which is suited for computer vision applications, in order to see if our new analysis actually helps improve this network.
Our research can also be utilized in a medical sense. The way a face paralyzes during a stroke is different from the way it paralyzes during Bell's palsy. Sometimes it's hard to see the difference between the two with the naked eye. Our model and DISC can be used to detect the tiny differences between the two types of paralysis.
In the future, we're also aiming to create a web app that people can submit videos to and receive their emotions back. And we're also currently working on implementing more efficient and effective recurrent neural networks and transformers to improve our current model and get more accurate results. I just want to say thank you to Rena Max, Jess Hoflich, Zikang Dong, Shifu, Dr. Paul Polak, and Dr. Miriam Rafailovich for this project.
As we have the question view graph, when that comes out, just shout out if you have any questions for that session. And if not, we'll go on to the next session that involves nanotoxicology of titanium dioxide, which was mentioned before. And in addition, also the ability to sequence DNA on a chip and a microcircuit and how to construct that. So please take it away.
All right. Hi, I'm Matthew, and I worked with Navia, Annie, Darshini, Harsha, Sherwin, Sarah, and Jeffrey in our study of titanium dioxide nanotoxicology. So titanium dioxide is used in a number of cosmetic products, but the small size of titanium nanoparticles can lead to penetration of the skin. We wanted to observe the impact of titanium dioxide on skin. So in order to do this, we grew skin-equivalent organotypics and treated them with titanium dioxide. In order to validate the results we saw in our initial analysis, we conducted numerous tests on both epidermal keratinocytes and dermal fibroblasts. Some of these tests were discussed in the previous symposium. These were flow cytometry, where we found about 50% association with titanium dioxide in keratinocytes and nearly 100% in fibroblasts, fibroblast cell proliferation and keratinocyte colony formation efficiency, which both showed that titanium dioxide made no significant impact on growth rate, and lastly, DAPI and actin filament staining, which showed no significant deformation of actin filaments. So these early tests seem to suggest that TiO2 had very little impact on the ability of fibroblasts to support the epidermis, which was consistent with our histological analyses.
We conducted immunohistochemistry tests by staining skin organotypics infected with TiO2 with filaggrin and corneodesmosin. For filaggrin staining, while the control depicted abundant amounts of filaggrin depicted by the red, less filaggrin was formed in keratinocytes treated with TiO2. Additionally, almost no filaggrin formed in the very aberrant stratum corneum in treated keratinocytes and fibroblasts A and B. We could therefore conclude that titanium dioxide had a profound impact on the differentiation through the regulation of filaggrin expression.
Corneodesmosin is a protein integral to localizing the stratum corneum at the top of the epidermis. The protein is found in the stratum granulosum, which we can see demarcated in red after staining for corneodesmosin. In both the control and TiO2 infected keratinocytes, this layer appears maintained, although the stratum corneum may form aberrant structures above it. With both keratinocytes and fibroblasts treated, we still see a distinct layer except for in image A. So we can conclude that TiO2 treatment doesn't have too much of an impact on corneodesmosin expression, and thus some regular cell dynamics may be preserved.
Elastic modulus is the resistance of a substance to deformation. In the case of fibroblasts, the higher the modulus, the stiffer, less elastic the cell. We checked how TiO2 concentration affects fibroblast cell moduli. The data showed a positive correlation between the TiO2 concentration and the cell modulus. Atomic force microscopy was used to run the test.
I'm sorry, I think we're supposed to be on the first slide. Yeah, sorry.
Yes, okay.
So in order to examine the effects of TiO2 on the ability of the skin to contract and remain flexible over time, what we did was we plated fibroblast-laden collagen into well plates at different various concentrations of TiO2. And over a period of 32 hours, we took images of the resulting gel as it contracted. Using image J, we analyzed the area of each gel as it contracted in the well plate for every pictured hour. And what we found was that despite some slight variations, which we think were probably due to human error in the plating, generally we observed that for each concentration, the collagen contracted at very similar rates. So we can conclude that the titanium dioxide did not significantly impact collagen contraction.
And for this part of the project, in the future, we will first repeat the collagen contraction experiment and analyze the data using this method to cross-validate our results. We will also conduct the same flow cytometry experiment that we did on fibroblasts with keratinocytes, as well as find an effective way to quantify the features of our skin construct, such as the thickness of different layers of the skin equivalents, to perfect our results of the skin histology test. And last but not least, we'll also test titanium dioxide's effects on angiogenesis of the skin, which basically is the skin's ability to form blood vessels.
Next slide, please. So another one of our projects was examining the impact of TiO2 nanoparticles on bacterial resistance for both fibroblasts and HUVEC cells. And we also looked at gene expression to try to explain the trends that we saw. So in terms of our initial testing of cell properties, we also wanted to see if TiO2 nanoparticles would affect cell infection by S. aureus bacteria, which commonly affects skin. So we quantified the infection for both HUVEC and fibroblast cells through increasing concentrations of TiO2, which you can see on the x-axis of these graphs. To quantify infection, we made colony counts from bacteria collected from the infected cells. And the trend for the fibroblast cells is that As TiO2 concentration increases, relative bacterial infection appears to increase as well. But for the HUVEC cells, we see the opposite trend.
In a previous study, a similar result to that of the fibroblasts has been shown in HeLa cells, where exposure to TiO2 reduced resistance to bacterial infection. This result was tied to an increase in cholesterol outside of the cell membrane by upregulation of the genes ABCA1 and ABCG1 in response to TiO2. Increased cholesterol aids infection because S. aureus, the bacteria used, requires cholesterol for membrane attachment. So we applied this idea to our experiment and hypothesized that since fibroblasts showed increased bacterial infection with TiO2, the cholesterol secretion genes would be upregulated, similar to the HeLa cells. However, our RT-PCR results showed that the genes were actually downregulated, suggesting that cholesterol is not the mechanism behind the increased bacterial infection. This conclusion leads us into our future work.
With future works, we will rerun fibroblast experiments with additional cholesterol blockers to confirm our results, and we will also repeat work with HeLa cells to confirm previous results known in literature. Finally, we would like to thank all our members and mentors for the guidance and support throughout the process. Thank you.
Thank you. And I should also make a small comment that these students created actually a functional skin equivalent in the laboratory with Professor Marcia Simon and the Living Skin Bank, where, as was mentioned by Isaac Cohen, these kinds of constructs can be used to test toxicology without animal trials.
Hi everyone. Our project was determining the efficacy of using transposase TN5 to cut DNA on a PMMA surface. So DNA sequencing is a very quickly growing field, especially with the rise of next generation sequencing technologies. And it has a lot of applications in health and medicine. So the process of DNA sequencing starts with cutting the DNA into sequenceable portions. And when cut in solution, DNA can lose its orientation, requiring complex computational processes to reassemble it. So our objective was to cut DNA into sequenceable segments on a PMMA surface with transposase, more specifically TN5, with the hope of holding its orientation. So transposase is an enzyme used to cut DNA, therefore it was used as our cutting agent. So our methods consisted of creating the TN5 and DNA solutions, dipping them onto silicon wafer spun cast with PMMA, imaging our samples, removing the TN5, imaging them again, and then analyzing our results with image J.
So once the TN5 is attached to the DNA strands, it cuts the strands, but the strands will not be separated until the TN5 is taking off the strands. Therefore, we tested the efficiency of using SDS soap solution and proteinase K as a TN5 removal agent and compared the results. We later determined that proteinase was a better removal agent. As shown in figure 1, before proteinase, the DNA strands are long and stretch out lines. After proteinase, the lines are fragmented into smaller segments, separated from one another in an ordered manner, which is the desired result because it is easier to sequence shorter strands of DNA if the order of the sequences are not messed up. Afterwards, the proteinase reactions were repeated for different concentrations of TN5 to find a trend. The results show that changing TN5 concentration does change the length of the DNA strands that are cut. The more concentrated TN5 is, the shorter the strands will be. As shown in figure 2, we are able to produce an average of 2 to 3 microns long of cut DNA strands using 5 microliter of TN5 solution. Well, when we used 20 microliter TN5 solution, we ended up with an average of 0.5 micron long cut DNA strands. And we wanted to give special thanks to Dr. Sokolov and Dr. Rafalovich for helping us do this project. Thank you.
Hi, my name is Hannah and my partners are Rachel and Darren, and our project focuses on the deposition of DNA onto thin film gratings. Despite significant improvements in high throughput DNA sequencing in the last 20 years, all current sequencing techniques are only able to generate reads less than a few tens of kilobases and have difficulty labeling and reassembling structural variations and repeats. Such constraints mean that much of the genome that has a role in gene expression and in disease is at risk of being lost through the sequencing. Therefore, our group hypothesized that DNA can be deposited onto flat polydimethylsiloxane or PDMS samples and then stamped onto silicon wafers with gratings to leave the DNA suspended across the troughs of the grating, which is called bridging, and also provide steric clearance for the transposates to simultaneously cut and label the DNA strands. We have also explored the idea of spin casting the DNA onto the gratings to achieve a similar goal. This method will allow for larger base pairs of DNA to be labeled and analyzed quickly with minimal risk of losing important genome and information and spatial organization.
We used two methods to deposit DNA onto gratings. The first method we used was stamping DNA onto silicon gratings, which is shown in figure 1. First, we placed a piece of PDMS that was dipped into solutions with different concentrations of lambda DNA in either DNase or TE buffer, with varying concentrations of sodium chloride to promote adhesion to the PDMS. Instead of sodium chloride, we also tried using a 1% concentration of glycerol to encourage the DNA to transfer from the PDMS to the silicon grating. The method we used to dip the samples of PDMS is seen in figure 2. We imaged our samples with a Leica microscope, and if the DNA appeared to be stretched out, we stamped the sample onto a PMMA-coated silicon wafer. The second method we tested was spin casting DNA onto silicon gratings. Static spinning and dynamic spinning were both analyzed. For static spinning, a droplet of DNA solution was placed at the center of the silicon wafer and was incubated for various lengths of time before spinning. For dynamic spinning, a pipette was used to deposit the DNA solution at the edge of the wafer while it was spinning.
Results indicated that PDMS that was cured in a 60°C oven for 2 days increased the adhesion of DNA on the PDMS. However, this made it difficult for the DNA to be stamped and transferred onto the silicon wafer. The salt also made it difficult to transfer the DNA, causing the DNA to rip. To solve this problem, glycerol was added instead of salt to aid transfer. Figure 3 shows one of the most successful results we've had. We dipped a PDMS sample into a solution with a concentration of 2.5 nanograms lambda DNA per microliter of DNase buffer with 1% volume over volume added glycerol, as shown by figure 4, and stamped the PDMS across an 8.6 micron linear grating for 15 seconds and no wait. We saw consistent amounts of bridging across the wafer. For static spin casting, altering the solution incubation times and spin speeds did not have effects on the results. For dynamic drops, a rotation rate of 5,000 RPM showed some bridging, but also caused breakage of the DNA strands as the speed was too fast. Overall, spin casting yielded adequate results shown by figure 5, but they were difficult to replicate. More research is required to find the preferable conditions for spin casting. We would like to thank Professor Sokolov for his guidance these past few weeks and Dr. Rafailovich and the Garcia faculty for their continuous support. Thank you for listening.
Hi, everybody. My name is Derek Days, and my partner is Om Patel. And we worked on trying to find the effect of SDS on DNA and if it could desorb or lift up DNA molecules from a flat surface in order to sequence genomes. Lifting DNA off a flat surface is important to next-generation sequencing, or NGS. NGS is promising for important fields like medical diagnoses, forensics, and gene editing. NGS relies on short-read techniques, or cutting a longer DNA sequence into shorter, more manageable segments. However, doing so requires a protein called transposase, which requires the DNA to not be attached to the surface, since the transposase needs to physically wrap around. SDS is an ionic detergent that can denature biological substances, meaning that it has potential to desorb DNA. The sample surface used was polymethyl methacrylate, or PMMA, spun cast onto a silicon wafer. This wafer was then dipped onto a 0.286% DNA solution, also containing dye, and then half of the wafer was dipped into SDS and re-dyed again. Finally, the wafer was submerged in a DNase buffer to make the surface hydrophobic. The resulting surface was imaged under a Leica 63x magnification lens.
Using those methods, Derek and I made 17 dishes of samples. As we imaged them, the results varied. Like the top image, some of the samples came out contaminated or too high concentration of DNA. And the clustered image, as you can see, was not okay. We had to alter our SDS concentration, our DNA concentration, and methodology However, some samples validate our goals. The image below shows the clear border between the DNA and SDS, indicating one of two options. Either the SDS successfully lifted off the DNA, or it removed the dye. And to further experiment, we re-dyed the samples again. And in the future, we will use a 1% SDS solution since that was the most effective. We used a column heater that has the potential to completely desorb the DNA, and we kept increasing the temperature, getting closer to that 90 degrees Celsius point, which would completely denate the DNA. So we approached that, but we never crossed it. And additionally, a new detergent, Triton X, has produced promising results in desorbing DNA. It quickly created a highly visible border between the Triton X and DNA in the first try, as the bottom image shows. And the magnitude of efficacy of Triton X warrants the need for future research. Me and Derek would just like to thank the Garcia program and Dr. Sokolov. Thank you.
There are no further questions to this symposium. The next presentation is something very, very special, put together by Evelyn Shu and her co-producers. And I was going to flash up from my phone the picture that said that it arrived to Jessica at 3:04 a.m. today. Hot off the press. Take it away.
cannot stop laughing.
Alright, someone just take that off. Take that off, take that off.
Thank you very much, Evelyn, for this. This was a wonderful little showcase of that the students in Garcia have fun together, and really emphasizes that forming lifetime friendships is really one of the important goals of the Garcia program. So now we continue to session 7, which is repurposing cellulose, and some research with Professor Stephen Walker on the longest living oral bacteria so far that we've seen. Tony, Elizabeth, take it away.
So in the first presentation of this section, we'll be listening to a presentation about 15-year-old samples of the bacterium Enterococcus faecalis, which is fairly common in oral infections. This will be presented by Andrew Zhang, Shreya Tiwari, and Michelle Bach, and they did their work under Dr. Walker of the dental school. The next two presentations involve deweaving cotton fibers and focuses on using environmentally friendly methods to do so. These projects were conducted by Ivan Yuen, Hannah Tao, Andrew Yin, and Alina Sheikh, and they were conducted under Dr. Michael Schifo.
Hello, we are Michelle, Shreya, and Andrew currently working with our RU, Tony Deluxe, and Dr. Walker on the research project of addressing the question of whether or not bacteria E. faecalis can survive for 15 years. E. faecalis is a microbe that can be often found in the dental plaque and the root canal, being a gram-positive bacteria with a thick peptidoglycan layer. Initially, the Stony Brook Dental School conducted multiple experiments to analyze whether bacteria E. faecalis can penetrate the sealant in the root canal before discontinuing their project and placing the samples in the incubator for nearly 15 years. Capitalizing on such known traits of vancomycin resistance or its thick layer, we utilized and designed primers such as the 941 primer to target the gene of interest and conducted multiple PCRs to prove the survival of the different strains of the bacteria. Continuing on, we also tested various detergent samples that could eradicate the spread of the microbe in the root canal, thus proving that E. faecalis is the longest living oral bacteria. Given that the previous record of the survival of E. faecalis was 12 months, the leap to nearly 15 years will garner further discussion to topics such as persister cells and vancomycin resistance.
So going a little bit more into detail about the specific methods we used, in a previous study, E. faecalis was identified in samples taken from root canals using polymerase chain reaction. So we used this method to confirm the identity of our samples in this study. To resurrect the original strains from the root canal study, samples were rehydrated with 1 milliliter of brain heart infusion broth and incubated at 37 degrees Celsius. We then ran PCR with a 16S ribosomal RNA primer to confirm for presence of bacterial DNA and confirm the quality of DNA for future PCRs. We ran 3 separate PCRs using 3 sets of primers. The first primer was a 310 base pair primer for the 16S gene specific to E. faecalis. The second primer was a 941 base pair primer for the VAN-A gene, which encodes vancomycin resistance in the bacterium. And the third was a 404 base pair primer for genes in the Entner-Dodorov pathway and alternate pathway of glycolysis used in E. faecalis.
So on to results and discussion. Table 1 gives a quick summary of our PCR results for the 3 PCRs that Shreya just mentioned. And as shown, all the results are very consistent, except for the 941 base pair primer in 2 of our samples, still strongly suggesting the presence of E. faecalis. Figure 1 gives a quick example of the actual results. And finally, a couple important sources of error to note. First, low molecular weight of the loaded samples caused overflow of DNA into adjacent wells in the gel electrophoresis. And second, DNA clumped together in the well for the positive control on the first day, causing an unwanted negative result. Lastly, conclusions and future work. So the PCR results we've received so far give a confirmation that E. faecalis is indeed present in our 15-year-old samples. And this is a significant result, especially given that the previous record for E. faecalis survival was just 12 months. For the future, we will continue work on ongoing biofilm assays, aiming to compare any phenotypic differences between the resurrected and the parent strains. Finally, we would like to thank Dr. Park of the Stony Brook School of Dental Medicine and Dr. Rafalovich, as well as Tony Deluxe and Dr. Walker for all of their hard work to make our research possible. Thank you.
All over the world, we've been seeing huge increases in cotton waste brought on by increased demand for cellulosic fibers. Many current methods for recycling cotton cellulose are costly and hazardous. To reduce the environmental footprint of recycling, we will show that mild physical and chemical treatments can deweave cotton muslin fabric into fibers with many applications. Diagonally cut fabric pieces were placed in a 40 milliliter solution of 0.5 molar citric acid, 0.5 molar sodium nitrate, or a 3-to-1 mixture of the two, and stored at 600 RPM at 50 degrees Celsius. We then dried all the samples.
We found that the deweaving solution was successfully reused 3 times, but significant decreases in solution volume, pH, and fiber bleaching were observed. Thus, the solution should be replenished with fresh stock solution during each reuse. Next, as seen in Figure 1, optical microscopy showed loosening of threads, disappearance of the weave pattern, and removal of contaminants after deweaving.
Using scanning electron microscopy, or SEM as shown in figure 2, we observe that the structure of the fiber bundles is mostly preserved. However, the fibers are also rougher after deweaving, pointing to fiber structure alterations that may impact mechanical strength.
The Fourier transformed infrared spectra, or FTIR, shown in figure 3, resemble cellulose spectra in literature and suggests the preservation of the beta-glycosidic linkage. Comparison across the spectra of the C double bond O peak suggests that esterification between citric acid and cellulose occurred, which we later confirmed through back titration. The FTIR data was also used to compute crystallinity indices, shown in table 1, which have no substantial changes after deweaving, suggesting minimal alteration to cellulose polymer arrangement. Tensile tests, seen in table 2, revealed that compared to untreated fibers, citric acid deweaved fibers have a lower ultimate tensile strength, but a greater elongation at rupture. This decrease in strength is likely due to physical rather than chemical changes.
Due to suspended cellulose microfibers, we observe negative zeta potential values in a deweaving solution. In the future, we aim to further clarify the deweaving mechanism and quantify the degree of esterification. Experiments will also be scaled up using samples of greater mass. Now we'll move on to our second project, which explores an application for our deweaved fibers.
Silver nanoparticles have well-studied antimicrobial and catalytic properties. Cellulose can prevent agglomeration and stabilize silver nanoparticles. So this study seeks to synthesize silver nanoparticles on deweaved cotton muslin fabric through the chemical reduction method. We used approximately 0.24 grams of fibers that were deweaved with citric acid and washed. We immersed these in a 20 milliliter solution of 0.01 molar silver nitrate and reduced the silver nitrate with 2 to 6 drops of 0.01 molar sodium borohydride. Then we ultrasonicated these samples at 40 degrees Celsius for 20 or 30 minutes and dried them.
We imaged the nanoparticles with SEM as shown in figure 1. Energy dispersive X-ray analysis, or EDX, was simultaneously performed to verify the existence of silver, as shown in figure 3. The fibers ultrasonicated for 20 minutes showed more and finer nanoparticles and less agglomeration. The size of the nanoparticles on both samples were found to be less than 40 nanometers in diameter.
We then performed zeta potential tests on the solution used for synthesizing silver nanoparticles. The 20-minute sample showed a zeta potential of negative 29.32 millivolts, while the 30-minute samples showed a zeta potential of negative 27.92 millivolts. These relatively high negative zeta potentials indicate significant electrostatic repulsion amongst the silver nanoparticles, which prevents agglomeration, and we observed this especially in the 20-minute sample.
Lastly, Raman spectroscopy revealed peaks associated with cellulose, silver metal, and silver oxide, as shown in figure 2. In the future, we'll perform dynamic light scattering to further characterize the size of nanoparticles. We'll also evaluate the properties of the silver nanoparticles through lead rate measurements, electrochemical techniques, including cyclic voltammetry and dye degradation tests. Now, we would like to thank James Quinn for the SEM, our mentors Dr. Chifo and Dr. Rafalovich, the Moore and Charitable Trust, and the Garcia Center for their support. Thank you.
And I should also say as a comment that this very nice and cohesive work done by this group under the guidance of Dr. Michael Chifo was really done across continents. Some of the students, Hannah, some of the students are here present in person, while the other half was collaborating with them from across the globe, and 12 hours of time shift. So amazing, amazing work, guys. Very nice. So now we move on to liquid embolic agents. And this is a collaboration with Dr. Chander. And the chair of this session is Megha Gopal from Stony Brook.
Hi, everyone. So brain aneurysms are bulges in the blood vessels surrounding the brain. A rupture in this aneurysm can cause life-threatening bleeding and even death in over 40% of cases. As such, there is great interest in developing methods to prevent such rupture through the use of medical devices that reside on the cutting edge of engineering, material science, and medicine. We have two groups who pursued this research today. Our first group worked to optimize a liquid embolic agent that could be injected into the aneurysm sac and gel within the deformity, keeping out blood flow and thereby preventing rupture. This is optimizing a minimally invasive, reverse thermoresponsive liquid embolic agent polymer treatment for brain aneurysms by Noah, Elena, Shahir, Doyan, Brianna, Evelyn, and Lorenzo.
Hi everyone, our project focuses on developing a gel treatment for brain aneurysms. We refine this gel by testing different concentrations of the main polymer F88DMA, as well as the contrast agent, cross-linker solution, which solidifies the gel, and other additives. This gel is reverse thermoresponsive, meaning it is liquid at lower temperatures and more gel-like at higher temperatures.
There were 3 parts to our project. First, we drew blood from a rabbit and combined it with 3 different cross-linkers to see which ones induced blood clotting. Second, we adjusted the concentration of different substances within our gel and tested how that affected the overall stiffness in vitro and in vivo. Third, we made cylinders out of different cross-linked gels and recorded which ones expanded the least when submerged in water.
In our first animal experiment, we tested blood clotting or thrombogenicity using 3 cross-linker combinations involving ammonium persulfate and various catalysts. The black spots that you see in the picture are blood clots, and the rightmost vial with the timid catalyst had no blood clots, meaning that it was not thrombogenetic and the safest for us to use. We thus exclusively used the timid cross-linker in our ensuing experiments. In the second animal experiment, we successfully injected a gel of 1.25% timid and 99 milligrams per milliliter iahexol into a rabbit and are continuing to monitor it.
Could you move to slide 2, please? Through rheometry, we found that gels with higher timid concentration of 1.25% and lower contrast agent concentrations of 99 milligram per milliliter concentrations took less time to cross-link and reach their maximum stability than their 0.75% timid 130 milligram milliliter counterparts. This is important because we want the gel to cross-link as fast as possible after injection and remain stiff so that it doesn't get washed away by the bloodstream.
We cross-linked 5 types of gels, all with different additives but otherwise identical compositions, and submerged them in water at 37 degrees Celsius, which is the approximate temperature of the human body. Figure 6 depicts the percentage increase in mass of the hydrogel cylinders 2 days after cross-linking. Since mass is proportional to volume, we assume that an increase in mass implied a smaller but similar increase in volume. Our results show that using 1% tetramer 107 as an additive may inhibit swelling, as it has a percentage increase in mass around 3% less than that of the control.
So overall, we've taken 3 large strides forward in this project by identifying a cross-linker that doesn't cause clotting, determining concentrations at which our gel will successfully inject and also remain in a live organism, and identifying an additive that prevents swelling and thus mitigates the risk of aneurysmal rupture and subsequent hemorrhage. This progress enables us to look towards further refinement and new areas of exploration.
In the future, our in vitro research will focus on optimizing the gel's biodegradability along with observing the effects that additives have on its swelling, healing, and micropore formation. Our goal is to improve cross-linking for future in vivo testing and usage. Our group would like to collectively thank and acknowledge Dr. Rafailovich, Dr. Sadasivan, Robert Wong, Aaron Slatsky, and Megha Gopal for their support and guidance throughout our research.
Our second group in the session worked to combine polyvinyl alcohol, a leading material in vascular graft models, with additives to promote endothelialization, or physiological integration with human tissue. Creating vascular grafts out of this material will allow for seamless integration of aneurysms into in vivo models, letting researchers better test medical devices. This is cell adhesive properties of polyvinyl alcohol and additives as potential vascular graft materials by Caroline, Sophia, Sohana, and Emma.
So currently there's a failure of small-scale vascular grafts due to thrombosis and intimal hyperplasia. To help combat this, our group decided to test PVA with additives such as gelatin and fibrin to find more reliable grafts that can also model aneurysms more accurately.
We started with the solution preparation. We created a 10% PVA solution in deionized water. We then added our crosslinkers, STMP, and NaOH, and divided our solution into samples to test our additives. These additives were 0%, 1%, 2.5%, and 5% RGP clay, 2% gelatin, and 10 mg per milliliter fibrinogen solution. All of our samples were sterilized before we began testing, and the clay was autoclaved. For our cell experiment, we cultured our fibroblasts and prepped 3 24-well cell plates, dedicating 3 wells in each cell plate to each of our samples. We did a media exchange, and then we plated our cells on top of the samples, fixed and dappy stained them to prepare for imaging.
So these images in the center look pretty similar across days, but these are all from day 1. On the top left, where cells were plated directly onto the well plate, there is a really high cell density and many cell connections. The top right PVA-only gel has more circular cells with smaller cell connections, and the bottom left gelatin gel is insignificantly different from the PVA-only gel. And the bottom right fibrin gel has higher density and longer cell connections, which is a promising result. The RDP clay gels showed no growth, so the oven-heated clay may have killed the cells.
We also conducted rheology tests to analyze the stiffness of the gels, and we discovered that the elastic modulus increased with the clay concentrations and that the gelatin gel had an elastic modulus similar to that of the 1% RDP clay. We also tested the thrombogenicity of our solutions by spin casting them onto silicon wafers with the polystyrene solution as a positive control. We added fibrinogen solution to half the wafers to compare results. And we found that fibers only grew on the samples with polystyrene as expected and not on any of the ones with clay, indicating that the PVA RDP clay solution is not thrombogenic, which is a positive result.
So in the future, we hope to spread RDP clay on a thin PVA gel to prevent clumping and then also hence prevent the RDP clay from sinking to the bottom. We would like to thank all our mentors for their continuous support, especially Dr. Rafailovich and Aaron Slotowski and Robert Wong. Thank you all for listening.
Thank you. Okay, there are no more questions. This work was done in collaboration with Dr. Chandramouli Sadasivan from the Department of Neurosurgery. And the last talk, the goal of the last talk was to make artificial vesicles and veins that had the same feel of real ones to enable surgeons to practice surgeries outside of the operating stage. Thank you. And the next symposium, Symposium 9, are polymer blends. And please take it away.
Hello, our group will be investigating the optimizing conductivity and strength of biodegradable polymer, graphene, nanoplatelets, nanocomposites via 3D printing. And the group members are Taven, Gavin, Gabo, and Horace. So electronic waste contaminates water and soil sources around landfills with toxic chemicals. Previous research has shown the viability of creating a biodegradable and conductive graphene polymer blend. And new such nanocomposites with higher conductibility, moldability, and 3D printability will be explored. Graphene is a conductive and strong allotrope of carbon, which we blended with isotactic polypropylene (IPP) and polylactic acid (PLA) at various concentrations. We hypothesized that the differential attractions for these polymers will lead to bicontinuous pathways for graphene, enhancing conductivity in a biodegradable manner.
First, we blended polylactic acid, polypropylene, and graphene nanoplates in a polymer blender. From here, we could either thermopress it, which gave us a rectangular prism, and then we could test the electrical conductivity using that. We could also 3D print a dog bone shape, allowing us to test the tensile strength. And we also printed 3D cubes, which we used the FLIR thermal camera to measure the thermal conductivity. We use a scanning electron microscope, or SEM, to examine the cross sections of each sample. Lastly, we measured graphene's affinity for either polypropylene or polylactic acid using contact angle imaging.
So this contact angle imaging did reaffirm our data and our hypothesis. As you can see, the affinity for isotactic polypropylene was higher than that of polylactic acid. We also looked at SEM data, and you can see on our 75-25 and 40-60 samples that we have some circular domains, which we hypothesize are PLA, and we hypothesize that surrounded by those are isotactic polypropylene, which has the graphene aligned and embedded within it. We conducted electrical conductivity data, and we could see a maximum on our 15% graphene graph at the 40-60 pp to PLA ratio, and this was about 1 × 10⁻⁵ Siemens per meters. At the 20% graphene sample, our electrical conductivity just kept increasing, and we did see a maximum around 1 × 10⁻⁴ Siemens per meters. We saw that with increasing graphene concentration, there was a significant increase in electrical conductivity, and this was similar for thermal conductivity. We collected relative thermal conductivity data, as you can see by our FLIR thermography section, and the thermal cube was able to allow heat to transfer more evenly throughout it when graphene was introduced. We hope to calculate specific thermal conductivity values in the future. However, when graphene was introduced, efficiency increased, but so did our brittleness. By our elongation graph, you can see that there was a very significant drop-off in elongation when graphene was introduced in the sample, which is going to be a major setback in processing and manufacturing of this blend.
There was a direct proportionality observed between the graphene and conductivity, which was comparable to silicon and gallium arsenide, essential components of semiconductors. We noticed a direct trade-off between efficiency and durability with increasing graphene, which is definitely something that needs to be considered for manufacturing. Our feature work consists of a full analysis of mechanical properties, specific thermal conductivity values, and transmission electron microscopy to explore IPP domains, as well as the general testing of more samples. The graphene polymer blend is a cheap and biodegradable alternative to existing electrical components with applications for developments in semiconductors and electronic systems. A special thank you to Yu Chung Lin for his constant mentorship and guidance, and Dr. Rafaelovich for providing us such a wonderful opportunity with the Garcia Summer Research Program. Thank you.
Hello, my name is Andrew Shin. I worked on the flame retardant polymer blend using biopolymers. Our goal of this research was to investigate which mixtures of PLA, RDP, APP, biopolymers, cellulose show flame retardants. And the way we created those polymers was first by melt blending the desired polymer. And we used the hot press to make a stick-like mold. Then we subjected it to the UL94 flame test, which consists of 2 10-second direct exposure to a blowtorch and a column below to check whether the column ignited or not. So onto the results. There are mainly 3 groups, the one with the biopolymer, one with RDP cellulose, and our flame retardant solution. The one with the biopolymer did not do as good, as you can see. Compared to just PL and RDP, it did better, as in the first trial, the polymer with the RDP and biopolymer didn't ignite, but in the second trial, the column still ignited, so the UL94 flame test grade didn't change. The second group was with the RDP cellulose and APP. We found that optimizing for like the least amount of APP, we found that 88% PLA, 8% RDP cellulose, and 4% APP still yielded a flame retardant polymer with the V0 grade. However, we did note that slightly changing the APP amount did make it not flame retardant, as you can see below the highlighted one with the 90% PLA that yielded a V2 grade. And the reason why we tried to minimize APP was, as you can see in figure 2 and 5, if we include APP, that significantly reduces the tensile strength of the polymer. So that's why we're trying to optimize the amount of APP so we can make a mechanically sturdier polymer. And we also created flame retardant solutions, which include distilled water, lactate monomer, APP, and RDP. We found that the solutions with APP and RDP, or just APP, had its column charged when the column was dipped into a solution and then subject to a blowtorch for 20 seconds. However, without it seems to have ignited, which reaffirms that APP and RDP does show flame retardants. We've also done a Raman spectroscopy on the char of the maritime solutions columns. The presence of the D band and the G band from the Raman shows the formation of disordered carbon structures. We also note in the FTR data, it can seem like the APP and RAP solution, or just APP solution, the presence of the phosphorus oxide position, the FTR, which shows char, which is what we want for a flame retardant solution. So in conclusion, adding biopolymers does improve the flame retardants only slightly. It doesn't affect enough to change the UL94 grade. And we also note that PLA, RAP cellulose, and APP shows flame retardants, but with the weakness of having a significantly weaker tensile strength. So in our future work, we intend to optimize for this tensile strength. And if we have more biopolymer, we intend to make more polymers and possibly compare the polymers with the RAP cellulose and APP. Shout out to grad student Yue Feng, the RU's Christian Owen, and Dr. Rafalovich for this Garcia program. Thank you.
Thank you. And if there are any more questions, the goal of both of these groups was to try to replace current petrochemical polymers that are non-degradable with biodegradable polymers that have similar specifications and properties. And both groups have made fairly significant advances in this area, especially in the last 2 weeks. So thank you. Our last session, session 10, really highlights our ability to have teachers involved and having Garcia both as a research but also as a teaching platform for students, and what is the best way to teach students new principles and collaborations. So let me introduce, before we begin, Mrs. Rebecca Issaroff, who was the science director of the Lawrence High School region, and she has been with Garcia for many years, including having been the person to negotiate an agreement with Materials Research Society, an organization for material scientists, to accept and make even a special deadline so that our students can participate in the MRS conference this fall. Mrs. Isra.
Thank you. Every year, the Garcia program, after they complete the safety training, they perform a group experiment to determine the molecular weight of a polymer by various methods such as spin casting, ellipsometry, and other experimental techniques. And this accomplishes two goals. First of all, it introduces them to how to operate in a lab. And the second goal is that it emphasizes collaboration, teamwork, organizational skills, and error analysis. And this is very important in any job, to be able to work as a team, to be able to collaborate. And due to our excellently motivated students this year, they have written a paper detailing this educational technique for publication. We're going to submit it to the Journal of Chemical Education for publication. And so I'm going to introduce now the group that has been spearheading this paper to talk to you a little bit about it. And then the next part is that we've had a couple of students who volunteered to do extra work in spin casting various polymer molecular weights in order to better understand error analysis and how to correlate their results with the known molecular weights. This will be submitted to the Materials Research Society as a scientific entry. So I'm going to introduce both first the spin casting paper for the Journal of Chem Ed.
All righty. Hello, everyone. My name is Alex Wang, and alongside Thomas Luong and Justin Kim, both of whom will be presenting with me today, Sean Fang, Arkajyoti Sinha, Sile Sood, and Hannah Tao, we've been working on writing a paper for the Journal of Chemical Education based on spin casting, the determined molecular weight project, which we worked on the second week of the Garcia program.
So hands-on learning has been shown to be one of the most effective forms of education in high school age students compared to other forms such as didactics and multimedia usage. So from this, diverging from that thought for a moment, determining molecular weight for a polymer is one of the most important procedures, as it greatly helps in understanding the properties of it, such as viscosity, chemical resistance, and recyclability. Therefore, us 90 Garcia students participated in a hands-on experiment to determine the molecular weight and molecular properties of various polystyrene samples through the means of spin casting. This allowed for many of us to get our hands-on experience in a university laboratory setting, which we did not have a familiar setting about, and we wanted to publish this experiment in order to allow many other high school students to have a similar experience and to kind of follow our steps through our process.
So before experimentation, the students received chemical safety training from the environment of the salt and safety of the brainwaves and sizes of reactors, constants and equipment such as
No, I can't hear. Sean, if you're speaking, we can't hear you.
I believe it's Thomas, but I don't know
Oh,
his
okay.
microphone is. I'm not sure.
Can you hear me?
It's very muffled. Are you covering it?
Can you hear me now? Should I
Yeah,
speak
much
louder?
better. Much better.
Okay.
No, you're good now.
Okay. So concepts and equipment such as PPE, fume hoods, and safety data sheets and chemical waste were also covered. Furthermore, students also received a facilities tour and were given example demonstrations of equipment used in experiments such as the Fourier transformed infrared spectrometer, and or the FDIR machine and the differential scanning calorimetry machine, or the DSC machine. So various experimental procedures were also utilized to test different polystyrene samples of known and unknown molecular weights, such as 35K MW polystyrene from Aldrich and Raman Cubs from Nissen.
Yeah, and throughout this experiment, one of the main concerns of high schoolers in a university laboratory setting is the hazard, because many of us have not had hands-on experience in the university laboratory before. However, throughout this entire experiment project, students were supervised by adults at all times and given sufficient prerequisite safety training, such as through the EHS presentations that we got in the first week. And also engineering controls were heavily emphasized by grad students and postdocs throughout the experiment, such as wearing proper PPE and also using the chemical fume hood properly when pouring toluene. So looking at the results of our initial project, glass transition temperature, molecular bonding, thickness, and critical concentration of the samples of polystyrene were determined. And a new universal curve is currently being determined, which will be discussed in the next presentation about extrapolating to the 2000 and 3000 angstrom thickness. Within the last section of our paper for the Journal of Chemical Education, one of the main things was really to emphasize how this can really be translatable into many different settings with high school students and other ages, such as undergraduate students, such as that. So we made student and supervisor handouts and wrote out all the specific procedures that we went through. And also we showed the verification of our results in the additional control data, which will be also discussed more in the next presentation. Thank you.
Hi, everyone. My name is Sean, and along with Arco, Alex, Justin, Sahil, and Thomas, we worked on studying the effects of monodispersity and polydispersity on wafer thickness after polymer spin casting. The polymer we are using to study this is polystyrene, as all of you should probably know. Polystyrene is harmful to the environment as it degrades very slowly and leaches toxins into its surroundings. Most commercially produced polystyrene contains multiple molecular weights of the polymer, so it is important to understand the theory behind polydispersed polystyrene in order to discuss the issues around recycling it.
We first studied monodispersed polystyrene using the molecular weights 30K, 90K, 123K, 200K, 400K, 650K, and 2000K. These molecular weights were spin casted at the concentrations 10 milligrams per milliliter, 15 milligrams per milliliter, 20 milligrams per milliliter, 25 milligrams per milliliter, and 30 milligrams per milliliter. We then used the ellipsometer to measure the thickness of the polymer coating on each silicon wafer. The results are graphed along with their lines of best fit in figure 1. We then moved on to polydispersed blends of polystyrene, which involved using different molecular weights in order to see the effects of varying molecular weights on the blend itself. So we blended 200K polystyrene with 400K polystyrene, 90K polystyrene with 650K polystyrene, and 30K polystyrene with 2000K polystyrene. These blends were spin casted at concentrations of 10 milligrams, 20 milligrams, and 30 milligrams per milliliter. And the resulting data, along with the respective lines of best fit, are graphed in figure 2.
Initially comparing the monodispersed and polydispersed data tells us that at low molecular weights, the polydispersed blends exhibit thicknesses that are greater than each individual monodispersed component. However, at high molecular weights, the polydispersed blends seem to approach the thickness of the higher molecular weight monodispersed component. In the future, we plan on producing generalizable equations that constitute a universal curve for monodispersed polystyrene, relating concentration to molecular weight at 2000 and 3000 angstroms. Establishing a concrete grasp of the theory behind polydispersed polymers and their deviations from monodispersed polymers will have exciting applications in recycling in our fight against pollution. We would like to thank Dr. Afalovich and the Garcia program for giving us the opportunity to conduct this research. We would also like to give special thanks to Dr. Jerome for his guidance. Thank you.
Thank you very much. And that was concludes the scientific part. And now a special presentation just describing the Regroup magazine and what it means as the conclusion to a scientific program.
Hi, so my name is Elizabeth, and I'll be presenting about Regroup today. So I'm here today with Quentin, Andy, and Hannah to introduce Regroup magazine, and I'll share my screen to the website. Okay, can people see my screen?
Yes.
Okay,
Yes.
cool. So last year, Nature editor Philip Ball gave a really impactful talk on communicating complicated concepts through these relatable, easy to understand metaphors. And we were so inspired by his words that a group of us Garcia kids got together and created a magazine to break down complicated, jargon-heavy research into engaging articles intended for a broad audience. We published our first issue, The Future of Science, last November, as you can see here. And this year, we've returned to the 2022 Garcia program. So some students from last year's program and a few students outside Garcia returned to produce a second issue with this year's Garcia students. Over the past 2 months, 35 students came together to brainstorm, plan, write, edit, and illustrate for the second issue. And today we unveil for the first time our second issue, Evolving, which focuses on biological evolution in tandem with the tiny steps of scientific evolution that compound into major breakthroughs. We are so excited to have many more editors, authors, and incredible original illustrations this year, including our cover designed by one of our illustrators, Erin Kim. So now I'll turn it over to one of our editors, Quentin, and I'll share the magazine.
Hi, I'm Quentin, and I'll be talking about my experience editing for Regroup this year. Our edition's theme was evolution, both in the context of biological evolution and scientific evolution, so we made sure that all articles tied into this theme. During the editing process, I worked with a few other editors on each given piece, and we focused on turning the submissions into digestible, easy to understand articles for the common reader. It was incredible to learn about groundbreaking research in topics from lactose intolerance to neurons to aneurysms, all while improving my writing and science communication skills. All in all, it was a really rewarding experience. Next, I'll hand it over to Andy, one of this edition's authors.
Hi, my name is Andy, and I'll be going over what it was like writing for Regroup this year. I co-wrote the article AI Crash Course with Ed and Hen, which should be coming up in a second here. And during the writing process, we especially focused on communicating through metaphors, such as comparing neural networks to a child's imagination. And I found it incredibly fulfilling to have our original idea come to life as an article, now featured in a finished magazine, which looks pretty nice. And it feels like a culmination of years of study and excitement, being able to produce a piece that can teach people about something that you're passionate about.
Very,
And
very
yeah,
nice.
we have a final slide here just for some final words. So thank you, Quinn and Andy, for sharing your experiences. And now just for this last view graph. So if regroup looks like something you're interested in, it's still not too late to join because we have a lot of exciting future opportunities coming up. So for example, we plan to expand outreach to get featured by science competitions. And you can also start a regroup chapter at your school to get students involved in high school research and create articles to submit to regroup. And of course, we still, plan to continue publishing more issues. And so if you want to stay updated on all these exciting things, please go ahead and check out our website, follow our newly created Instagram, and join our Discord server, all shown on this slide. And if you need the links to any of those things, feel free to contact any of us via Discord, email, anything like that. So thank you so much for listening, and congratulations to everyone on carrying out such a successful symposium this year.
Thank you very much. And for final concluding words, I cannot think of a more fitting conclusion to the Garcia Program Symposium. And I can say I did not see this last presentation at all before, but it really personifies everything that the Garcia Program stands for. And that is to create the community of the future scholars and scientists, whatever profession they may want to go to, as long as they have respect for each other, collegiality, respect for science in every discipline, working together in collaboration. We are in very good hands on our future to you. Thank you very, very much. And that concludes the Garcia 2022 Symposium for this year. I'll see you all again as we start getting prepared for the MRS abstracts next week. Okay, and so begins our trek into the future of science. Thank you very much.
Thank you.